Thank you so much for showing up. We're going to see a couple more people dribble in here, but while they are, I just wanted to say thank you so much for coming to this session on the future of science and Healthspan and all the technologies. We've been hearing a lot about how biotechnology and gene editing is going to create profound changes in our way to treat the issues associated with longevity and Healthspan. But this session's going to be completely different. We're very fortunate to have Dr. Sagriva. She has some really incredible research that's really not being done anywhere else on the planet that she's going to be sharing with us. She comes to us from Tufts University. She's also a researcher at Harvard University at the Wyss Institute. Myself, I'm Zach Lynch. I'm one of the founding managing partners at Jazz Venture Partners. We're an early stage venture capital firm based in San Francisco. We have about $1 billion under management. We invest in a lot of things around the convergence of science and technology and how that impacts human performance. Clearly, Healthspan is within our mandate. We've made lots of different investments. After Elena shares with you the brilliant research that she's doing, I'll have a few minutes maybe to share a couple of those companies. But why don't we kick this off, and Elena, why don't you take it from here?
Thank you for introduction. Hello, everyone. My name is Elena Sergeyeva. Thank you. I work at Tufts and Wyss Institute at Harvard with Michael Levin. So today I'm going to talk about innovative approaches in aging field, specifically about bioelectricity, morphogenesis, and aging. Yeah, so I want to start with favorite question in the field of aging. What is aging? But before that, I want to talk a little bit about this thing. So the definitions which we give, they should not limit us. They need to empower us. What does it mean? They need to allow us new frameworks and tools to look at the questions, to look at the problems, and to solve that problems. So regardless what aging is in the field, it's damage accumulation or whatever, that's all fine, but we need to look at additional aspects which are very often overlooked. So in our approach, aging is a defect of morphogenesis and morphostasis. Morphogenesis is easier. Like morphos is a form, so this is creation of the forms and structures. And morphostasis, if you're familiar with homeostasis concept, that means that the form will be homeostatically maintained over time. So for us, aging is the process when cell collectives fail to maintain pattern, homeostasis, and keep up with structure and function all the time. So, but what patterns we're talking about? I want to ask questions, where do anatomies, which probably disintegrate through the time with aging, coming from? So we all start our life as quiescent oocytes, and then this oocyte starts to divide. There are many cells there, but somehow we are not ending up as a bag of undifferentiated cells. We have very differentiated organs, tissues, structures, body parts. How is it happening? So DNA, which we usually consider to be a software of life, just specifies proteins, the building blocks of our bodies. But where is anatomy coming from? So how do cells know what to build? And most importantly, when to stop? So they grow limb, they stop with 5 fingers, they don't grow 6. They don't go very long limbs. So they stop at the necessary time point. So why do cells sometimes defect from target morphology? For example, when we deal with some developmental disorders, or when we have malignant growth, which we call cancer. So this is basically deflection from target morphology. And how to repair and remodel this damage, including how to repair and remodel this damage which is happening with aging, right? And so we also can push this question further and ask what engineer would ask. Is it possible to build some other forms with given default genome? And we usually think, okay, if we have perfect DNA without damage, without mutations, we will have perfect body, which is not aging, doesn't have diseases, disorders, but it's actually not complete truth. There is a striking example in nature, and it's not a single example. This is just the most interesting one I want to provide here. So planaria, this is flatworm. It's very smart, very cognitive. It has very differentiated body with all the systems, like nervous system, like even some analogies of brain, and so on. And this worm is highly regenerative, cancer resistant. It doesn't have cancer at all whatsoever, and it's immortal. Some forms of planaria, they're immortal, so they basically maintain their body for like millions of years. And they have the messiest genome around. How is it happening that such a bad, messy genome can produce such perfect body form, which can maintain its structure over many, many, many years? So it seems that should be some higher level of organization with some emergent properties and additional competencies which can overcome these genomic problems. And here I want to give you an analogy of nervous system, and analogy of computer science. So you know hardware and software. So we think that hardware in the body is genome and its building block proteins, the proteins which are built based on our DNA. And software is actually like something on the top of that. I will explain now what. So let's take analogy with nervous system. So we all know that our nervous system consists of synapses which are connected, which exchange some signals, electrical signals between each other, and form electrical circuits. And these circuits, electrical circuits, this is hardware. But there is something more interesting happening on the top of that hardware, which is electrical dynamics. And this is completely known and accepted in neuroscience, so that there is this next level of regulation, this electrical dynamics, which form memories, which is basis for our memories and learning and any cognitive functions. But this nervous system didn't emerge from nowhere. It's actually evolved from normal somatic cells, which also have all the same properties like ion channels, electrical synapses. They exchange electrical signals between each other exactly like neurons would do. And they also have this layer of competencies on the top, this electrical dynamics, which you can see here on the right side, which we call bioelectrical pattern. This is voltage, membrane voltage across cellular collectives and tissues. So again, neuronal system didn't happen from nowhere. It actually evolved from communication between normal somatic cells. It's evolved for speed, it's evolved for precision. Yeah, but the truth is that all cells in our body communicate using bioelectricity. And this communication pattern forms so-called bioelectrical pattern, which we consider to be software on the top of genome and protein hardware. So, and how do we detect these bioelectrical patterns? On the left here, you can see this is frog Xenopus laevis embryo, which is developing. And while it's developing, it changes its bioelectrical pattern, which can be measured using advanced imaging techniques. Like we use special voltage reporting dyes, and we can put it under microscope and measure that bioelectrical patterns and see how embryo is developing and how it changes patterns while being developed. And also, we can model these bioelectrical patterns. And these endogenous bioelectrical patterns, they actually depict the structures of the body which are going to be formed. So you can see here, it's again developing Xenopus laevis embryo. Oops. Sorry. Yeah, and here on the right side, it's a face. So this is like facial anatomical structures, which you can see as bioelectrical pattern, which is developing along with that anatomical structure development. And when anatomy deflects from its target point, which we call cancer, it's also reflected as changes in bioelectrical pattern. Yeah, so, and we can not only measure and detect these bioelectrical patterns, we actually can manipulate that. So, and for this, we use tools of neuroscience. So the patterns, they are properties of membrane voltage, and membrane voltage is formed with ion channels, with some work done by ion channels and gap junctions. And these ion channels and gap junctions can be manipulated with drugs, like ion channel drugs, and also we can express ion channels in cells by some tools like injecting RNA or some kind of gene therapy. Yeah, so with this we can manipulate, change bioelectrical pattern, and see if we can, with changing bioelectrical pattern, we actually can change formation of body structures. Yeah, and we have such examples. So here you have a striking example of induction of an eye with manipulation of bioelectrical pattern. Eye is a highly complex organ. And for example, you want to create eye in the back of tadpole, frog embryo again, so because frog embryos with our model organisms. Yeah, so if you think like molecular biologist, what you would do? You would probably come up with a list of 100, maybe 1,000 of genes which are responsible for eye formation, and then you will try to change the genes, like overexpress some of them, downexpress other genes, and then like manipulate that in timely manner. Good luck with that. So it's just not possible. So it's not possible to make an eye on the back of embryo by manipulating genes, but what can we do? And we have this done experimentally. So we can inject RNA, which expresses certain ion channel in the cells. And then what's happening? These cells start to form, you see they are here in blue, they express this ion channel, they start to form eye structure in unusual place in the tadpole. And it's not only that cells form that structure, but they recruit other non-modified, non-genetically modified cells to do that work. So they are basically like you give short prompt this RNA signal, specific ion channel, and then cellular collectives start to do their job. So they make the eye. And we have other examples also done in our lab. So this is not only induction of the eye, this is also induction of fins, induction of otocytes, induction of complete beating hearts with just manipulating certain ion channel expression. This is also expansion on forebrain and induction of complete correctly patterned ectopic limbs. So you see here, this is extra limb in a frog, so which can be induced just by expression of certain ion channels, by manipulating that bioelectrical pattern, which is software of life. So, and we not only can do like strange Frankenstein things in the lab, but we also can do very useful things like regeneration of the limb. This is again frog, our favorite model organism. So with bioelectrical prompt, with expression of certain ion channels, we can induce regeneration of the limb. And now this work is being done with mice. So Disclosure Morphoceuticals company, spin-off of Mike's Living Lab. So they're regenerating limb in mammals now using the same paradigm. Yeah, so, and also what can we do? We can rewrite anatomical pattern memory. So we can change bioelectrical pattern this way, that we can rewrite memory, this pattern memory which exists in the body. This is planary again, and make it to do 2 heads instead of 1. So this is like, this is not existing in nature, but if we manipulate patterns a certain way, we can grow 2-headed planaria, and if we cut them a half, they tear apart, they start to regenerate the whole worm. they can end up as two-headed pulmonary again. So we can change pattern memory, and this is true memory because it can be represented in next generation of the worm. Yeah, so all the examples I showed you here, development, birth defects, traumatic injury, regeneration, cancer, and now aging, you can see that all these examples are actually solving the problems of information processing in morphospace, in the space of anatomical structures, right? And we ask the question how to control emergence and scaling of cellular activity toward desirable complex system-level outcomes. So, and what about aging more specifically? So we can measure bioelectrical patterns. Here you can see example of frog embryo, and this is Hydra, and also we can measure bioelectrical patterns in other model systems like organoids, tissue explants, and hopefully future in bigger animals and whole animals. Then we can put this pattern against of age. So many of you know this aging clocks, right? The most of them are epigenetic aging clocks or transcriptomic aging clocks, but we do bioelectric aging clock. So we basically see how this bioelectrical pattern in model organisms changes with time. So, and what we preliminarily see that it corrupts with age, so it becomes more fuzzy. So cell-to-cell communication is not so sharp and precise as it was before. And then we can not just detect and build with bioelectrical clocks, we can also reprogram by using that ion channel gap junctions expression or manipulation with pharmacological approaches. We can change this bioelectrical pattern, and as you saw that with that examples, we can, like changing patterns, we can change body structures and functions. Yeah, so what we learned here in the lab. So cellular collectives need to know the correct pattern to create and maintain body structures. Anatomical maintenance is guided by pattern memories, which are stored in bioelectrical circuits. We can analyze and manipulate cell collectives using the tools and concepts of neuroscience. And aging is due to that cells forget the correct bioelectrical pattern, and cells remember the pattern, but lose ability to execute, right? And now we take the science to the next level. I also presenting here Astonishing Labs, which is spin-off of the work which is done in Mike's Levin lab. So Astonishing Labs is a venture platform company that identifies and commercializes discoveries done by Dr. Michael Levin and his team, and creates innovative therapies that change medicine. How do they change medicine? So what I showed you, this Levin's analysis of the operating system creates multiple novel platforms for drug discovery and development. So basically, this is a paradigm shift how we look at drug discovery and development. So what is current mainstream approach? We manipulate genes and proteins. We basically rewire hardware as it was done in IT industry in 50s. So we manually try to rewire the hardware by manipulating genes and proteins. But when IT revolution happened, it happened when we learned how to use software, how to execute very complex programs, not by rewiring computers manually, but taking advantage of programmable software layer. And we believe that revolution in biotechnology, in medicine, will happen the same way when we learn how to manipulate software of life instead of just rewiring genes and proteins. So bioelectric drug discovery and development has very unique advantages. So first, we use novel therapeutic targets. This is operating system instead of micromanagement. Also, bioelectric drugs, then they're prompts, they're basically prompts, if I can use analogy of machine learning and AI. So we give a short prompt, and then as you saw with that eye induction example, then cellular collectives start to do what they're supposed to do, right? And this does not require chronic treatment, and this is very good, particularly good for aging interventions, right? Because we're not going to take anti-aging drugs whole our life. We probably want to have something like a prompt, then our body can then use that prompt to change itself. And here, the most important thing here is biology is the ultimate problem solver, not drug developers. So we make biology to solve the problems. So this is basically like idea about Astonishing Lab. I will just give you a short overview. So Astonishing Lab is very different from standard VC approaches. So it invests in long-term studies. So it doesn't look for immediate revenue, like in 5 years returns, but it finds very early stage works and starts to invest in very early stage, basically a lab work stage or even conceptual stage. And then this is like what we call it very patient funding. Yeah, and this is our team. Some of you might know Mark Siegel, who is also investor in X Prize Longevity, and other remarkable people here. And if you have any questions about Astonishing Labs, you can use these contacts. Yeah, and big thank to all the people who done the work I presented today. Of course, Michael Levin, our collaborators, and our funding support. So Astonishing Lab, Templeton Foundation, DARPA, Paul Allen Foundation, NIH, Sloan Foundation, and others. Yeah. Thank you.
Dr. Sergeyeva, that was so impressive. This is a fundamentally new paradigm around which you can actually begin to sort of control aging. How far have you guys taken it so far?
So you saw that examples which we did in the lab with regeneration, for example, or induction of organs. So this is all amazing laboratory work, yeah. But we also try to take it into clinic. For example, I did disclosure about Morphoceuticals, Morphoceuticals company. So they already regenerate limbs in mammals and mice. So normally, you know that limbs normally do not regenerate. They already try to do that like in startup company, basically. Yeah, so but what about aging? We should look at aging as at all that things like regeneration, development, cancer. It's basically a continuum of different processes like body. What's the main thing here? Body has to upkeep structure and function all the time. If it doesn't do the job, then we have cancer, developmental disorders, we cannot regenerate, and we also age. So these are basically all processes in the same line. So, and from that, you could look at regeneration spin-off as a precursor for our future aging.
So if you go out like 5 or 10 years, where would you see potential bioelectric drugs being developed?
You mean?
Across all spaces. Do you
all
think
spaces, okay. Yeah, so it's already being done for regeneration, for cancer.
Cancer?
Yes, cancer. And yeah, the next spin-off will be in aging.
And can I ask a really basic science question? How do you actually figure out how to stimulate and how much to stimulate a particular cell? Mm-hmm. Does that take practice or like,
okay.
How does that happen?
Yeah, it's a great question. So we don't use any external electrical fields. We don't stimulate cells with electricity. What we do, we prompt cells. So we let cells to do their job. So we just give prompt as some drug which would change, like tune the ion channel activity, so to speak, and then cells start to like do the job better, so build structures, maintain structures better. I can give you one example with cancer. So we'll look at cancer as, okay, some cell starts to grow, and then we end up with tumor. From our perspective of bioelectricity, what is cancer? So all cells, they form cellular collective to build some structures, like they have common goal. But what happens? One cell starts to treat other cells which are around that cell as environment when it becomes cancerous. Why it's happening? Because this cell forgets how to talk to their peers, so this cell stops to communicate with neighboring cells properly. This is like problem with bioelectrical communication, which we study, and then it becomes cancerous. So, and what is our approach here? We return the cell into cellular collective. We don't assault on that. We don't try to kill that with chemotherapy, radio, or so on. We're just trying to give proper prompt to improve this cell-to-cell communication with bioelectrical drugs. And then we showed experimentally in the lab that model organisms, they remove cancer when we use that bioelectrical approach. Yeah, so hopefully next thing will be to do that in humans.
That's great. So I know you all just got like a PhD in bioelectricity. What questions do you have? Simple, complex? I thought I saw one right there, yes.
Can we give me a microphone?
Is there anything being done in mammals? Is your first question.
You mean with bioelectricity? Mm-hmm. Yes. Yeah. Great questions. So bioelectrical pattern is measured using imaging techniques. If we want to measure bioelectrical pattern in model organisms, we need to measure that using imaging. Hyperpolarization, so we need to maintain that bioelectrical pattern, which would be the most suitable for whatever we want to do with that, cellular collectives.
Thank you.
So building off of that.
Can you use that so others can?
Oh, hi. Sorry. So building off of that question, for your toolbox in your lab, what is, you have hyperpolarization drugs, depolarization drugs, and I heard RNAs for
Yes.
ion channels. How many ion channels are you messing with in these things? Is it like 5? Is it? Yeah, I'm just curious.
Okay, in case of the most of examples I showed you, for example, ion induction, it was just single ion channel.
That's amazing.
That's amazing, yeah. But also we have machine learning platform being developed in the lab where we can find out which channels, which drugs are the most suitable for whatever outcome we want to have.
Any other questions out there? There you go. Right there in the back there.
Hi. Yeah, thank you so much. I'm wondering, it seems to me like aging is a more difficult challenge than some of the other things you're doing because you don't need an eye to grow in a certain part of you to not age. What you need is to have everything working together. I'm wondering, how do you think about that kind of more global quest and how you would restore kind of globally that bioelectrical homeostasis in the whole organism?
Yeah, thank you for your question. It's experimental thing, right? At this point, we cannot say that fighting aging in some of our model systems will be a more difficult task than, for example, regeneration of limb or induction of the eye. It's experimental question we have to test.
Thank you very much for this inspiring presentation. I have a question. Are there external disruptors of bioelectricity, like magnetic fields, static electricity,
Yeah.
and how can we access the information about how badly it disrupts our body and how to protect it, and is it possible to give up some things like mobile networks and etc.?
Yeah, two things here. Great question. Two things here. Biology is remarkably resistant to external fields because just we evolved in external electromagnetic fields. So, and if it would be disrupting to us so easily, then how would we maintain our bodies? How would we live and function? So biology is actually very resistant to external fields, so I cannot speak for phones or other devices we use. We never tested that in our lab. So yeah, but what I can tell you that it's really difficult with external fields. It's in experimental settings. It's really difficult to change bioelectrical pattern. So cells and cellular collectives, they actively push back if you stimulate them externally. But we don't use for that tasks, which I showed, like regeneration, cancer, and so on, we don't use external fields. This is all manipulating intrinsic bioelectricity. So we use drugs, we use ion channel expression, and it's not the same as it would be applying of external electrical fields. Huh? Probably, yeah. We don't study that.
Thank you.
And then one last question back there. Yeah.
Hi, yeah. I'm just wondering how precisely do you have to target and deliver your sort of ion channel interventions, etc., to be able to achieve your effect? Do you need to be quite spatially precise, and how does that translate into, translation?
Yeah, good question. So the difference of our approach from standard mainstream approach when genes are being manipulated is that we are not micromanaging our cells and cellular collectives. We don't tell them precisely, okay, this cell, you express this channel, you express that, and then you depolarize, you hyperpolarize. The beauty of our approach is that we're using prompts. So we don't have to be very micromanaging and specific to each cell. We just give a prompt, and then cellular collectives just start from there and do what they're supposed to do by themselves. So we like leverage intrinsic ability of cellular collectives to build.
Well, there you have it, the future of bioelectricity and health span and longevity. I was going to ask you a couple of questions, but I think you all did such a fantastic job of digging in here. Really appreciate you showing up, and continue to enjoy the show out there.
Thank you.
Okay, hi everybody. Thanks for coming to my talk today. Thanks to the organizers for putting together such a fantastic program. So I'm Alan Cohen from the Columbia Aging Center at Columbia University, and I'm going to talk to you today about trying to move away from thinking about aging as the target of our research and towards health, or intrinsic health more specifically, as the target for our research. So the motto from Hevolution that we've seen everywhere is, we are catalyzing the shift from lifespan to health span. I couldn't agree more, but I think there's a big problem here, which is, how do we define health? What do we mean by this word? So in order to get to this goal, we first need to answer the question, what is health? We also then have to figure out, how do we measure that health? Because for this to be a meaningful statement, we actually need to be able to measure the health. And then we also want to know how that relates to aging. So today I have 3 objectives. My first objective is to convince you that the best way to target aging is actually to target health itself. The second is to define health, and the third is to get us towards a framework to develop operational metrics of health. That's what I'm going to try to do today. I'm going to spend about 30 minutes on my talk trying to leave plenty of time for questions from the audience. So why is aging a challenging target? I'm going to go through several different reasons why I think aging is a hard thing to measure and target in an intervention sense. The first is that there's no consensus on the definition or nature of aging. So 2 recent papers have shown this. We have our paper here from Mechanisms of Aging and Development about 5 years ago, and then just a few months ago, this paper from Vadim Gladyshev came out. Both of them took surveys of a number of leading aging researchers and tried to say, what do we think aging is? Do we agree on any of the fundamental questions? And in both cases, here's one figure from each paper, we see that there's really extreme differences of opinion on even the most fundamental questions. So for example, in our paper here, you can just see these colors going from strongly disagree is the red, strongly agree is the blue, and in every single one of these questions, you see people at very opposite ends of the spectrum of what they believe. I'm not going to go through each question, you can just see visually how much disagreement they are, but these are questions like, is aging programmed? Or do we agree on what the nature of aging is? Or will we be able to dramatically shorten lifespan, right? Now, if we move over to Vadim's paper here, we see similar things. Is aging a disease? Some people strongly agree, some people strongly disagree. Is it essential for the field to have a consensus definition of aging? Strong disagreement. Will the average lifespan be increased a lot in the next 20 years? Disagreement there. What about stopping smoking? Would that count as rejuvenation? Disagreement. So even these kind of fundamental definitional questions, 2 major surveys of aging researchers have now shown that we don't agree on much of anything. So the one thing that we do agree a bit more on, though, is that aging is multidimensional. So if there's one thing that we can agree on, well, these are the 2 top questions from my survey where you see more agreement on one side than another. And that is that aging cannot and should not be measured by a single metric because it is multidimensional and heterogeneous. Most people were on the agreement side of that. Aging proceeds uniformly across tissues. Most people are on the disagreement side of that. Those were the 2 most consensual things, still not fully. Or we can look at, this is Dan Belsky's beautiful paper from the American Journal of Epidemiology in 2018. He looked at telomere length, 3 epigenetic clocks, and 3 biomarker-based metrics of aging, and showed that across these 3 different categories, there's almost no correlation in these metrics of aging other than with age itself, right? So we have a lot of different ways that we can measure aging, and they're not correlated with each other, right? So aging is clearly multidimensional. And then we can add in some more problematic aspects. So what about something like presbyopia? Presbyopia, nearsightedness, is caused by protein aggregations in the lens of the eye. As far as we know, this is not linked to other hallmarks of aging. I can't think of any good definition of aging that would not include presbyopia. It happens in everybody. It's kind of inevitable decline with age. I think any reasonable definition of aging should include presbyopia, and yet we're not going to get a hallmarks-based definition that is going to cover that. So any definition we would like would probably want to include things like that, like tooth wear. What about gray hair, which is maybe not causative, maybe not that interesting in that sense, but is nonetheless something that changes with age. So the next big challenge we have is that aging is heterogeneous. within individuals and across individuals. Here's an example of heterogeneous aging within individuals. This is work that I'm conducting with Nam Ki Hong, led by Nam Ki Hong from Yonsei University and Steve Cummings at UCSF. And Nam Ki is doing muscle fiber histology. And what he can show here, this is a muscle fiber from an older individual and a younger individual. You can see visually just how different they are. They decompose this into the individual cells and they get a Mahalanobis distance-based metric of the heterogeneity, and you see this clear increase in muscle fiber heterogeneity with age. It's one example of many where we see this kind of increases in heterogeneity. We see it, for example, in gene expression in the brain, we see it in ECG signatures, we see it in body composition signatures and blood biomarker signatures. Across many hierarchical biological levels, we see this increase in heterogeneity. That's within individuals. We see the same thing across individuals. So here is a 2014 paper from my group looking at inflammaging, and we see that there's a huge range at all ages of how much inflammaging individuals have. This is in Italy, but that range seems to be getting bigger with age as well. And here is another paper by Luigi Ferrucci and George Cashel looking at the increase in heterogeneity in glucose and oral glucose tolerance test results. So once again, we're seeing increases in heterogeneity across individuals with age. So the fourth major challenge in terms of developing metrics of aging is that aging involves trade-offs. So we need to figure out how to deal with these trade-offs. What do I mean by trade-offs? Well, if we look at the hallmarks, many of them are actually cancer protection mechanisms. Cellular senescence, telomere shortening, stem cell exhaustion, inflammaging, all have certain roles in protecting against cancer. Same thing with immune function. So many of the hallways also, particularly the nutrient sensing pathways, modulate immune function in ways that are probably designed to balance competing risks of the organism. So I would argue that many of the things that we consider as major targets for aging are things that are helping maintain the balance and competing priorities the body has in balancing those competing risks as best the body can do. For those of you who just saw Elena's great talk, I think she's arguing, and I would agree, that there's a lot of evidence that the body knows what to do best, and only in certain circumstances can we intervene and do better than the knowledge that evolution has accumulated over billions of years. And then the last thing is that many aging mechanisms of aging are compensatory or adaptive, right? This ties into what I just said, and we can't yet distinguish them well. I see Vadim here who has worked starting in this direction. I don't think we're fully there, but I think we are going in the direction of perhaps distinguishing them, but that's a major challenge. So if we want to think how do we measure aging, then we have all these measurement issues. Do we integrate across all dimensions? So we know that aging is multidimensional. We don't yet know what all the dimensions are. Do we have to map every single dimension and include them all, including the ones that are perhaps more important or less so? Do we have to know how important each dimension is in each context? What about the pathological, compensatory, neutral dimensions? Do we want just the pathological dimensions to be part of our aging score? What about things that cause aging, things that are effects of aging? How do we distinguish those? These are not questions that have a clear answer. There's no biological answer to that. We just would need to come to some agreement on what we mean. But because we don't have that agreement, knowing how we get there is a challenge. If we decided how we want to integrate the dimensions, we then have to think about how we're going to weight them. Are they all going to get equal weights? That's a decision. Do we give some of them more weights than others? Do all aging mechanisms have the same impact on the aging process in all contexts? If I live in one environment, one mechanism may be more important than in another. Does this mean that we need a separate measure of aging for each context that somebody is living in? So, and then lastly, how do we consider hierarchical levels of biology? Not just the cellular molecular level, but tissues and organs, and how everything integrates across all these levels. So I don't see this as an easily solvable set of challenges. It doesn't mean we can't find ways to measure parts of the aging process well. It doesn't mean we can't come up with something that we call a metric of aging, but I don't think we can easily get to something that will be a consensus definition that covers everything we mean when we say the word aging, and that will get at what we're hoping to do through targeting aging. So what I'd like to do now is to introduce you to the notion of intrinsic health, which I think will be a more tractable target for interventions. This is work that we've been developing with the Science of Health group at the Columbia Aging Center. I co-lead this group with Martin Picard. You can see our members here. We've got biostatisticians, complex systems experts, experts in population health, measurement of aging, and also in developmental health and pediatrics. We start with a complex systems view of life. So in this view, all of the different molecules in our body, arranged in different systems, influence each other in complex networks that try to maintain a global balance. So if you want to figure out what this molecule here is doing to this one here, you can't just look at these two. You need to consider that this molecule is going to affect this one, which affects this one, which affects this one, which affects this one, which is going to have the opposite effect now here. And that means that predicting what the system is going to do becomes a major challenge. So it's kind of biology 101 to know that we are a complex dynamic system, but this has been hard to incorporate into how we think about the biology. The next layer that I wanted to add on to this is that we're not just a complex dynamic system. We're a complex dynamic system that has undergone billions of years of natural selection that helps optimize the system. And that makes us very different from an economy or a weather system or other complex dynamic systems. And in particular, it's DNA, which gives the conservation of information over billions of years that allows this optimization process to happen. What is getting optimized? Well, evolutionary fitness. Fitness is something that happens at an individual level in a certain sense. We measure an individual's fitness. But in order to reproduce and achieve that fitness, you first need to do one basic thing, which is to hold your body together, to keep it from dissolving into its environment. And that's where the idea of dynamic equilibrium comes in. The only way that we stay alive is by having complex networks of molecules that interact with each other in this hierarchical structure that adjust to changing environments. Everything has been fine-tuned over billions of years, basically for that task, which once achieved, then we can layer on reproduction to think about the full evolutionary consequences. So we're starting with that model, and then we're thinking of health as kind of the center of that model. So health is the state when everything is going well with that dynamic equilibrium. And as things get lost in that system through injury, through aging, through disease, it spirals out. These three things could, and perhaps others, could interact with each other in various ways. But because there's so much heterogeneity in how things go away from the health state, the ways that we lose the health are harder to target than the health itself. There's a central homeostatic state to the ensemble, which, if we could figure out how to measure that, is going to be something that is more coherent and stable than the ways that health gets lost. Which brings us to our more global model, because we don't want to just think of health as biology. We know that there's a lot of things that are important to us in the context of our health that go beyond the basic biology. So what we've done is to make a model where we separate out what we call intrinsic health, which is the biological side, from realized health, which is health as we live it, as what it means to us. Can we be and do the things that matter to us? And so all of the biology gets filtered through what we call the lens of context. Our environment, affordances, social context, culture, values, perceptions. So for example, if I'm in a wheelchair, that, because I have perhaps some problem at a physical level, that's going to have an impact on my health that depends on whether I live in a place that has lots of ramps and lots of accessibility. If I live with a lot of stairs, the impact on my well-being is going to be substantially greater than if there's a lot of ramps to make things accessible. So we really think that this realized health is crucial to consider in terms of what things mean to people, but that is not a biologically targetable endpoint, right? The intrinsic health is perhaps biologically measurable, biologically targetable. So I think it's really important to separate out these two, knowing that they're going to have a relatively complex relationship. To show how this separation of intrinsic health and realized health can bring some clarity to certain notions, we can think of the example of aging. So we'd probably all agree that aging represents a decline over time in intrinsic health. It's linear in this graph, it doesn't have to be, but there's going to be some kind of decline over many decades in intrinsic health. But if you have the right contextual factors, you might continue to be very happy and satisfied with what your health is allowing you to do into older age. I think this is what Hevolution would like us to get to. They'd like us to optimize the intrinsic health, but then also preserve that realized health with the right cultural context as well. Or we can think about an acute challenge, an acute infection or wound healing, which will represent a decrement to realized health. Something temporarily goes wrong and then you get better, particularly if you have the right contextual factors. But this is a challenge our bodies are built to deal with. My intrinsic health isn't compromised just because I get a cold. My body's built to handle that. So I would see intrinsic health as staying quite stable over the course of any acute challenge such as that. So now we're ready, I think, to consider what intrinsic health really is. Where does it come from? And I would argue that this is like a first principles view of life itself. What does it take to make an organism? If we think of an organism as a complex dynamic system that's trying to hold itself together in the world, the first thing you need is energy. If the organism is full of lots of information, all of the non-randomness that is an organism, the only way that you can keep that from dissipating is with inputs of energy. This is what we know from the second law of thermodynamics. But energy is not enough. There needs to be communication in the body that keeps all of this going. The communication at many levels, many types of communication. There are neural signals, there are all kinds of circulating signals. There's signaling within cells, across cells, at many different hierarchical levels. All of that communication is essential to help the organism constantly make the right decisions to maintain its dynamic equilibrium. And lastly, none of that happens without a particular structure. DNA is a particularly structured molecule. There's a particular structure to each tissue and each organ. I have 2 arms and 2 legs and a head that are placed in a certain way on my body. So all of this communication and energy, we can't think of it happening randomly. It only works in the particular context of the structure of the organism that it's in. So the 3 of these things we think have a dynamic interplay that creates an emergent property of intrinsic health. We can't simply add up communication, energy, and structure and sum them to intrinsic health. It's going to be something that emerges from the ensemble. We think the perfect analogy for that is an electromagnetic field that emerges from the energy of a battery, communication with a wire, and a particular structure of a solenoid. It's impossible. So we know a magnetic field is real. We can measure it. We can use iron filings. This is not something in our imagination. This is not, magic. But at the same time, it's not something that you can measure or really understand by breaking it down into its component parts. Right? And so we think the same thing happens with health. When the right aspects at multiple hierarchical levels of energy, communication, and structure work together right, the organism as a whole functions well. And that is what emerges as intrinsic health. So now I'm going to draw a parallel here because I've just told you that we have communication, energy, and structure, but you can't add them up to get intrinsic health. So how would we measure it if we can't simply add up the pieces? And that's where I want to draw a parallel with evolutionary biology. So with evolutionary biology, we think of fitness as kind of the main currency of what's going on. This happens to individuals, which try to perpetuate the genes or the lineage over evolutionary timescales. And we break it down to measure it into survival, reproduction, age at maturity, and so forth. These are what evolutionary biologists call fitness components. And the fitness components, they might trade off with each other. I might invest in survival at the cost of reproduction, invest in reproduction at the cost of survival, but more or less if I'm measuring these things, I'm getting a good idea of what the overall fitness of the organism is. And fitness is actually pretty hard to measure, but fitness components are a much more tractable target for measuring. I think we can draw a lot of parallels here with intrinsic health, where rather than measuring at the individual level, we're measuring simply a moment in an individual's life, which can change over time. But it has the prime directive to perpetuate the individual over time, over individual lifespans. And the components here are going to be things like robustness and resilience, plasticity, performance, and sustainability. And I want to drill down a little bit into what each of these is. There's a lot of discussion about exactly what we mean by robustness and resilience. I don't want to get into a debate about distinguishing those two right now. But here I'm going to define them jointly as the capacity to maintain a given state or function in the face of external stressors. So I'm not looking at when you need to change your state, did you do it? I'm looking at can you keep a state when you want to keep it there, or if it deviates, get it back there. That's robustness and resilience. Plasticity is when you need to change your state, because organisms, unlike other complex dynamic systems, want sometimes to change from this state to that state. So can the organism get there when it wants to, and then also get back? That's plasticity. Performance is the capacity to effectively execute specific functional capabilities. This could be a wide range of functional capabilities, things that the organism is built to deal with in its historical environment. And then the sustainability is basically, can you keep all the rest of it going? You don't want to do all that very well for just a day. You want to do all that very well for a long period of time. So the sustainability is the capacity to maintain robustness, resilience, plasticity, and performance over long time scales. So this is not necessarily an exhaustive list of the components, but I think it's a pretty good starting point for a set of components that we could break down and start to try to measure as part of intrinsic health. So with all of this, we're now ready to give a formal definition to intrinsic health, which is a field-like state emerging from the dynamic interplay of energy, communication, and structure within the organism, which gives rise to robustness, resilience, plasticity, performance, and sustainability. So how are we going to go about trying to measure this more concretely? I'm not going to give you an answer today with here's the measure, but I'm going to give you a framework for how we're going to try to develop that measure. What we have at the bottom here are the pillars: energy, communication, and structure. Like I said, we're not going to try to measure these things directly, but they might inspire conceptually how we're going to think about metrics we can come to. I'm going to give you some examples in a minute. Then we have intrinsic health as a whole with its various components. We can come up with proxies for the components, or perhaps proxies for the whole ensemble. And we're going to use it to generate what I call a basket of proxies. We don't know which ones are good yet. We don't know if we've got all of the right ones, but we're going to just try to get some proxies there. And then we're going to try to statistically integrate them. There's lots of methods we can use, like causal representation learning, latent variable models, principal components-based approaches. But the general idea is that as we get more proxies, we're going to get more signal, particularly if we have a diverse set of proxies. But we're going to have diminishing marginal returns. We don't need 1,000 proxies. As we start to get to 10 or 20 or 30 of the right ones, perhaps we're getting close to a good measure of the overall signal. And then we're going to integrate that into some kind of intrinsic health metric. This is an example of what a latent variable model might look like. This is certainly meant to be just a kind of a caricature rather than a full example. We have intrinsic health, which would be determined by robustness, resilience, plasticity, performance, and sustainability. We could break each of them down further. So performance, we might have physical and cognitive and immune. And then the immune we could break down. And obviously this is just a partial diagram of what we'd be looking at, but it's a model for how we can start to think about how we could measure this. So what are the different proxies for intrinsic health and its components? A lot of them we have already. Heart rate variability, homeostatic dysregulation, GDF-15, which is a good measure of energetic stress. And I think this is a good example of where the pillars are conceptually informing what we're going to do. So as we understand that energy is an important part of what intrinsic health is, we can look at this marker, which is increasingly coming out as a stable indicator of bad things going wrong during the aging process, and understand that it's a marker of energetic stress in the organism. And so that this single molecule, it's not going to tell us everything we need, but it might be an important part of this ensemble, representing more of the energy side of things. And we have performance measures like cardiac function, kidney function. We could have a long list, right? And so all of these are things that we might start to put into our basket of proxies. We're currently in the process of developing transfer entropy metrics, proteomic dynamics of stress. Again, for those of you who saw Elena's talk, we have like bioelectrical conductance or bioelectrical patterns, which might be using the communication side of things. How well is that bioelectrical communication happening? Is that something that we could feed into a metric of intrinsic health? And then more generalized resilience indices. So for those of you who are interested in resilience, I'd argue that resilience is kind of the low-hanging fruit to get a long way towards intrinsic health. It's not the only thing we want to measure, but if we can get a good way towards measuring resilience, we're getting a long way here. So more specifically, what we're doing with this right now, this is a diagram representing the data that's been collected in the mitochondrial stress brain and epigenetic study, or MISBE, which is led by my colleague Martin. There are 70 healthy controls, and then 40 individuals with severe mitochondrial disease. When you have these severe genetic diseases, you have lose energy, but your health is just broadly compromised along many dimensions. So what we have is a model of looking at people with and without levels of intrinsic health, where we can then put them through various stressors, including the awakening response, 5 different stress protocols. We get multiple blood samples across this, or saliva during the awakening response. And now we're running omics panels on these saliva and blood samples so we can look at the dynamics of this. We can look at heart rate, blood pressure, respiration rate as time series across this. So we're doing time series analysis and trying to say, what does the signature of health look like in all of these deep data that we can get on these individuals? And so we're just starting these analyses. In some cases, for example, we only have pilot proteomics on the awakening response, but I'm going to show you that even with the preliminary data we have, we already are getting strong signals. So this is the transfer entropy data, where we can see the control individuals and the mutation individuals. This is during the recovery phase after the Trier social stress test. And we can see that for 4 out of our 6 transfer entropy metrics between heart rate, blood pressure, and respiration rate, we're seeing that greater communication between these signals is associated with being in the healthier state. This is looking at the proteome dynamics of just 6 healthy and 6 unhealthy individuals. And we looked at it 2 different pipelines to try to extract a signal. In this case, on the right, on the left, what we're seeing is 6 control individuals, and we said, with all 3,000 proteins that we measured change between baseline awakening and 30 minutes later, how do those correlate with each other? So 2 proteins that correlate well would be dark blue. We ran a cluster analysis, and we can see the patterns of how the healthy individuals correlate. Then we look at the unhealthy individuals, and we say, do we see the same thing? And the answer is not at all. The blue patterns here are completely different in the unhealthy individuals from the healthy individuals. When we choose 6 random individuals, and then look at the other 6, now it's certainly not perfect, but we're recovering some of the dark blue areas, indicating that even with a very small sample size, we can pull out a signal without training towards health that is showing that healthy and unhealthy individuals look quite different, whereas random sets of individuals look more similar. Another approach we used was to look at elasticity, which is elasticity is the ability to change from baseline when you and then come back down, right? So we're looking at both. Did you change a lot, and then did you come all the way back? We do that for all 3,000 proteins, and we then use t-SNE, which is it's unsupervised, meaning we're not trying to get it to show us who's healthy and unhealthy. Nonetheless, the first 2 axes here clearly distinguish the healthy and the unhealthy individuals, so that basic health state is one of the most important things that emerges from how these proteins change and come back during the awakening response. So this is just preliminary to show you how much potential there is to look at these deep dynamic data to get a basic signal of what health is. Where do we go from here? Well, I think the first thing we want to do is we want to fill that basket of proxies. We want to get as many different potential proxies as we can, and not all of them are going to work, but we're going to try to fill it up as best we can and figure out in subsequent models what works. So we're going to try to test for integration. Is there a single intrinsic health? Do we need to define several dimensions of intrinsic health? We don't know that yet. Then we're going to refine the model and repeat. And then here's the crucial part. Once we get a relatively good metric of intrinsic health, what I just showed you is very hard to measure in an individual. If you go see your doctor and you've got to go through 5 different stress tests and get 8 blood samples, nobody's signing up for that, right? So what we'd like is from a single test at the beginning, or from like a wearable, to get some signal of that ensemble. So once we get like DNA methylation or some other platform that gives us deep information from a single blood sample, we can train it to predict this thing here, so that we don't need this complex measurement protocol. We're also not forgetting the realized health chunk of it. So we're also in the process of developing metrics for realized health. So we need first to define a set of functional domains: cognitive, physical, social integration, vitality, disease status, what have you. We want some kind of integration where we're going to weight by preferences and environment to generate an overall metric of realized health. This is not as far developed as the other yet. John Beard is the one who's leading this effort with our group, but this is certainly work that is underway as well. And we do think that with survey-based data, we're going to get to a place where we can have integrative measures of realized health as well as intrinsic health. And we want to use those for different things. So the vision here is that intrinsic health will eventually become a single integrative measure, which can be measured from a blood sample or a single blood sample and or wearables. It could be an outcome for many health interventions and a metric of population health. And then intrinsic health would then be an appropriate target for aging interventions. Because if we're able to target intrinsic health, we should prevent many chronic diseases and improve well-being overall. We've got very low risk of side effects, trade-offs, and unintended consequences. And it's conceptually simpler because it's the homeostatic norm. So we don't have to worry that we're hitting the wrong thing as much. And so I'm coming back to my initial argument that the best way to target aging is going to be to target intrinsic health or some similar concept. And that's what I have for today. I hope I've got some good time for questions here. Any questions? You have
Yeah, it's very interesting model. So, just a simple question first. Like GDF-15, why is it energetic stress?
So GDF-15 is, during the integrated stress response, what's released from a lot of cells, happens during cellular senescence, but not only during cellular senescence. And so all the information we have suggests that what it does is it gets sent to the brain. There's specific receptors in the brain, and that after that what happens is there's a lot of regulation of various processes to try to conserve energy at the organism level. So, this is some of Martin's recent paper on the brain-body energy conservation theory of aging, relies a lot on GDF-15 and some other integrated markers. But the idea here is that even something like sarcopenia might not be fully pathological. It might be an effort to conserve energy when energy is in short supply.
It correlates really well with like disease status, like across diseases and mortality. So one of the best kind of proteins. So it's very interesting. Could you clarify how you view aging? Like what is your kind of definition of aging, and how is it related to intrinsic health? Just stress.
So my view of aging is that it's the loss of information in an organism. I don't think it's possible to have a biological definition of aging that is more circumscribed. It's not a biological process, it's a physics process that any complex dynamic system over time, unless it has a perfect error correction code to restore itself, is going to lose information. Now, David Sinclair has argued for the information theory of aging. Where I disagree with him is that he's thinking mostly at the epigenetic level. I think it happens at every single level. So if I lose an arm, I've lost information about the three-dimensional structure of my body. Every little cut I get that doesn't heal perfectly is a loss of information. Every loss of a neural synapse is a loss of information. Every change in the ratio of proteins in a certain cell that gets it away from its homeostatic norm is a loss of information. And so I think the ensemble of the ways that we can lose information is just intractable. Some of them are more important than others, but there's just so many ways we can lose information. And unless we find ways to restore all of it properly, we might make some progress in partially restoring the information, but I think it's a major challenge to restore information. And therefore, I don't think that radical treatments for lifespan are likely to work that well. I think that what's likely to work is preventing declines in intrinsic health by maintaining good conditions. Colin.
Thank you. Wonderful model. I was wondering how tipping points going from health into a disease state, how do they emerge from your model, or how you envision them emerging from your model?
Yeah, I mean, it comes back, actually, it's good Vadim just asked the question on my definition of aging, because as information gets lost, it's a vicious cycle. So I don't see it like necessarily a particular tipping point, but it's an acceleration of the process of loss. As more information is lost, the system is less well able to keep the rest of its information, and that loss accelerates. And then the tipping point would be death, essentially, the collapse of the entire system.
Just to stay on this, if it's a continuum, right, then we have a problem, because if there's no clear tipping point going into the disease, how can you then start fixing it?
So I think for certain diseases there may be tipping points. I don't mean to say that for no disease is there a tipping point for that disease. But I think that, like, if you look at the aging process or frailty, there's an acceleration where you can maybe see something of an inflection, but I wouldn't say it's a very, very clear tipping point.
Yes, thank you for your speech. I will be a bit provocative, sorry.
Please.
Yeah, so at the moment, the best way to know about healthy lifespan is still lifespan, you know?
Is what? Is lifespan?
Yeah, it's still lifespan, because as far as I know, the time between between a non-healthy lifespan and death is always about the same, except maybe for neurological diseases. Then there you can live quite long with in a very unhealthy life. But okay, the problem is to measure healthy lifespan. It is quite complicated. To measure lifespan, it is quite easy. In most cases, you know when you are dead or not. And more important, we want to make progress. And to make progress, we need tests on animals. And it is even less easy to know about healthy animals. So is this new, well, not so new idea of healthy lifespan not a bad idea?
Okay. So, I mean, yeah, I guess what I would say is, I think I've just shown you that I don't think we can measure intrinsic health well yet. And I think we need to be working in that direction. I think measuring aging is a challenge for all the reasons I've said. I do think we have measures, though, that get us something of the way there. So like some of John Beard's intrinsic capacity measures. So I think we have things that, depending on the situation, we can use. And it's a question of, is the use case appropriate for what we have now? I certainly don't subscribe to the idea that just saying you're disease-free or counting up chronic diseases is what we really want to get at. I think we want something deeper than that in terms of what we mean by health and health span. But I think it's very much a work in progress. And so in that sense, I agree that right now we don't have something that fully does that job.
I continue for animals. Quite often you have no articles and they say, yeah, the animals don't live longer, but they were healthier. How serious is that?
Yeah, I mean, I do think depending on the animal, you may have some good indicators of this. For example, if you're looking at monitoring in a cage and activity levels, and if the activity levels don't go down as quickly, that would seem to me, one good indicator perhaps among many. I'm not an expert in that animal literature, but I think there are things that you can do.
Thank you very much for the very interesting intrinsic health status that you determine. I was wondering, to which extent is it complementary or actually the converse of a frailty index determination? So frailty indexes are also input of a lot of different information from many different aspects. So to which extent does your health, integral health status, differ or be complementary to the frailty index?
I think the two will be inversely correlated pretty strongly. But my hope is that this is also a metric that we can start to use at other points in the lifespan. So for example, during childhood. So in points of the lifespan where the frailty index isn't giving us much sensitivity to pull out differences, I would hope that we're going to have more power to do that with these kinds of metrics. I would imagine that later in life there would be tight correlations.
I think you did a great job encapsulating where we're at today, and so it was good to actually see it all together. Let's take it a step further. Let's do a thought experiment. We've accomplished this. We're at this place where we've got this measurement. What are your hopes and vision once we're there?
Yeah, I mean, my hope is that this changes everything about how we think about medicine, right? So rather than trying to target specific diseases, we'll still have some need to do that, but with that preventively, you go to see your doctor, and your doctor is, from a young age, helping you monitor your intrinsic health overall, perhaps specific dimensions of intrinsic health, and seeing that this dimension is not going well, and coming up with personalized treatments to help you keep your intrinsic health over the life course. Yeah, so a frame shift.
Thank you very much for the talk. So thank you very much for the talk. So a background about me, I'm a rheumatologist. I'm a clinical research immunologist. So my question is, how does your definition fit into the quality of aging? So the quality of aging is very variable among individuals. So how would your definition explain it? And what would you think about this being an externally driven process versus internally genetically programmed process? Thank you.
Yeah, I mean, I think for the internal versus external chunk, we know that heritability of aging is relatively, heritability of lifespan is relatively limited, right? So I see this as mostly not a genetic problem. It's mostly an environmental problem that's getting us towards all the heterogeneity and how this is going to work. And not just in lifespan, but in who's going to age how. I think that's mostly an environment problem. The first half of your question was?
How does your definition fit into explaining the quality of aging variability?
Yeah, so that's where I think the realized health comes in. Because what matters to somebody is not necessarily their intrinsic health. What matters is the realized health, and that's going to be highly variable across individuals, but for reasons that are beyond the biology. And so, I'm a biologist, that's not as much my purview, but I think it's crucial to be considering, and that's going to be very different from one place to another and from one person to another.
Thank you.
Other questions? Okay, thank you, everyone.
processing. Now, an important consideration, as I said, is that aging leads to a reduction in brain glucose utilization. You can call it brain hypometabolism. And we have a very, I wouldn't say straightforward, but relatively common technology to monitor glucose utilization by the brain is fluorodeoxyglucose positron emission tomography. So you can actually determine where and when glucose is utilized by the brain. And it turns out that, and this, for example, is just a very large study that was conducted showing a decrease in glucose utilization in overall, but of course there are some hubs, like in particular frontal and temporal, regions where this glucose is utilization has decreased. But also relevant for the discussion today is the fact that there is actually a positive correlation between glucose utilization and decrease in glucose utilization and decrease in cognitive performance, which in a way is not so surprising because, as I said, glucose is used mainly, and you will see how, to maintain the excitability of neurons and therefore information processing. Now, the interesting thing is that this hypometabolism, the decrease in glucose utilization, is actually a particular metabolic pathway of glucose utilization that is decreased. And that is what is called aerobic glycolysis. I will tell you in a moment more about it. By the way, a metabolic process, and I will tell you more about it, which is primarily operated by glial cells, non-neuronal cells. But anyway, you can see here in yellow a high aerobic glycolysis in young adults, frontal, again parietal, and in old, even cognitive normal at a certain early stage subjects, there is a decrease in this glycolysis. You can see here the oxidative phosphorylation, the use of glucose, here remains quite stable, but what really dramatically decreases with age is aerobic glycolysis. So what is aerobic glycolysis? Just a brief reminder of glucose metabolism from the early college years. Glucose is phosphorylated to glucose 6-phosphate. It can then proceed through glycolysis to pyruvate, which is then oxidized in the Krebs cycle and oxidative phosphorylation. But there is a pathway which is called aerobic glycolysis, which is the production of lactate in the presence of oxygen. And then there are two other pathways. One is storing glucose as glycogen, which is obviously very active in liver in particular, and also the pentose phosphate shunt that actually produces reducing equivalents. So aerobic glycolysis is the production of lactate in the presence of oxygen. Now, where does aerobic glycolysis take place in the brain? Actually, in these non-neuronal cells, glial cells of a particular type called astrocytes. As you can see, these are star-shaped cells, therefore their name astro. And and this is something we discovered in the 90s. In the 90s, they are the main cell that produce energy for neurons. And we then and others showed that they are important to contribute to neuronal plasticity and memory, and are important in the prevention of neurodegenerative diseases. And you can already appreciate here, this is a very old drawing from Santiago Ramon y Cajal, where you see that this astrocyte that he drew had actually processes on the blood vessels, showing that actually they are the first entry gate of glucose into the brain. Now, astrocytes, as shown here, you see their interaction with or association with blood vessels. There are other glial cells, the microglia, which are actually the resident macrophages in the brain, very much involved in inflammation, and the oligodendrocytes, which are their main function is myelin-producing cells to produce myelin to cover the axons. We wrote a book with Yves Agid a few years ago that summarizes the role and function and properties of glial cells. Now, going a bit deeper in the relationship between astrocytes and other elements of the brain, you can see here that actually astrocytes have processes that cover almost entirely the intraparenchymal capillaries, the source of glucose, and they actually have other processes that ensheath synapses. And these areas here, they express receptors for neurotransmitters and reuptake sites. So this means that astrocytes are ideally positioned to sense synaptic activity when neurons are active and to couple this to entry of energy for the activity of neurons. And in fact, again, in the 90s and early 2000s, we described what is now known as the astrocyte neuron lactate shuttle, whereby neuronal signals like glutamate, for example, the main neurotransmitter in the brain, through a series of biochemical reactions that we characterized, imports glucose into astrocytes, and they convert it to lactate through aerobic glycolysis. They provide lactate to neurons through specific transporters, convert it to pyruvate, and then pyruvate is oxidized by the mitochondrial function in neurons. So there is really a close metabolic cooperation between astrocytes and neurons. Now, this means that we have to kind of have a paradigm shift and realize that most energy to neurons is provided by lactate through astrocytes. But the interesting thing is that lactate is not only an energy substrate, but we and others showed later that actually lactate is involved in memory consolidation. In fact, if one blocks the shuttling of lactate between astrocytes and neurons, one blocks memory consolidation. And interestingly enough, the lactate in this case comes mostly from glycogen, which, as I mentioned earlier, is stored only in astrocytes in the brain. This was a paper published in Cell a few years ago that really was a paradigm shift because, in a way, you have a very high function of the brain memory being dependent on a glial cells, which was for a long time considered glue. There is also evidence that lactate, through mechanisms that we are now elucidating and also others, actually regulate the expression of a number of genes, in particular those involved in neuroprotection and synaptic plasticity. A very recent addition to the signaling notion of lactate is a process known as lactylation, an epigenetic regulation like methylation or acetylation. So lactate is definitely not only an energy substrate, but also a signaling molecule. So in summary, what emerges from many years of research in many labs, and mine was one of the first to lead the way in this, is that astrocytes respond to neuronal signals, glutamate or noradrenaline, for example, that eventually through aerobic glycolysis or glycogenolysis produce lactate, and then this lactate is involved as an energy substrate, is involved in neuroprotection and neuronal plasticity. So, by the way, just in concluding this very first part, actually, energy is one component, but inflammation is always important in aging and degeneration, and this is carried out by another type of glial cells, the microglia. So, the question is as follows. Aging is a major vulnerability factor for Alzheimer's disease, as well as actually other degenerative diseases. And aging, I just told you, is, and we know now, is characterized by hypometabolism. So one way is to target brain hypometabolism in aging and neurodegenerative diseases. Why? We heard a lot about brain health today, and indeed, energy availability is absolutely necessary to maintain brain health. Why? Because it actually provides the resilience necessary to protect against, for example, oxidative stress or other insults to neurons. Now, if there is less energy available, so if we are in a condition of hypometabolism, then the threshold for resilience is actually decreased, and neurons become much more vulnerable and prone to neurodegeneration. So actually, recent clinical studies actually tried to address this with repurposing some molecules. For example, rasagiline, which is a monoamine oxidase inhibitor, which turns out to increase glucose utilization, has been used, for example, in a 24-week study to show that it decreases, if one decreases the decrease in glucose utilization over 6 months, because rasagiline promotes glucose utilization, actually there is a decrease in cognitive decline. And you can see here in blue the decrease in glucose utilization in light blue over 6 months, minus 4-5%. In the presence of rasagiline, the decrease is actually much less. And this is accompanied by what is called directional changes in the quality of life and cognition. So if you decrease or slow down the decrease in glucose utilization, you slow down the cognitive decline. Another study, also published recently, another molecule, riluzole, actually increases or maintains glucose utilization, so it slows cerebral glucose metabolism. You can see here the decrease in glucose utilization in control, and here with riluzole, you see there is almost no decrease over 6 months. Well, there is also there a correlation that maintaining glucose utilization maintains cognitive performance. And a very recent paper came out a few months ago showing that if in animal models, restoring glucose metabolism in astrocyte in animal models of Alzheimer's actually rescues cognition across various models of Alzheimer's disease, in particular the tau and amyloid. And so here again you see what we described many years ago. These are astrocytes prepared from iPS cells of Alzheimer's patients. Actually, this is the control. You have a hefty glycolysis, production of lactate, provision to neurons. Now, in the pathological astrocytes coming from Alzheimer's patient, this glycolysis is much decreased. And in this case, they identified a molecule, an inhibitor of the conversion of tryptophan to kynurenate that maintains glycolysis, maintains lactate production, and maintains cognitive performance in these animal models. So there is another actual vulnerability factor for Alzheimer's in addition to aging, and you probably know is the ApoE4 genotype. And in fact, these subjects, the carriers, there is about 13-14% of the population who carries ApoE4. About 40% of Alzheimer's patients carry the ApoE4 genotype. And interestingly, well before any clinical symptom for cognitive impairment, subjects with the ApoE4 genotype have already deficits in glucose utilization as monitored by PET. And interestingly, again confirming what I just showed you, in the case of ApoE4, actually astrocytes from humans with ApoE3 genotype, the most common one, have this type of glucose utilization profile in vitro. The ApoE4 is much decreased, and interestingly, the ApoE2, which is rare but confers protection to Alzheimer's disease, have a very solid uptake of glucose. So the question is, one should target brain hypometabolism to prevent not only Alzheimer's disease, but other diseases such as ALS or even depression are characterized by this hypometabolism. And so my lab started a few years ago to try to identify molecules that would essentially act on the astrocyte neuron lactate shuttle. In other words, act on astrocytes, boost their capacity to take up glucose and to produce lactate. And the idea, if one boosts this process, is that one could restore healthy brain metabolism by increasing glucose utilization and lactate production, improve neuronal functional resilience, and hopefully demonstrate in animal models of neurodegeneration positive effects, or even in aging in animals. And I'll show you some data that come from a spin-off from my lab from EPFL, where I am the scientific co-founder, so I declare a conflict of interest here. The company has identified a new molecule, original molecule, GP119, which actually boosts glucose uptake into astrocytes by acting on an astrocyte-specific molecule, enzyme, sorry. So it promotes glucose uptake and lactate production. And very interestingly, in a delayed time, maybe between half hour to 1 hour, it increases the expression of glucose transporter 1 at the membrane. And GLUT1 is the glucose transporter present on endothelial cells and on astrocytes, so the gateway of glucose into the brain. And this molecule, given orally, will stimulate glucose uptake, you can see here, in a dose-dependent manner. This is measured by brain biosensors in the prefrontal cortex, or lactate production, but also using FDG PET, the actual biomarker that is used in the clinics to monitor glucose utilization in humans, and you can see that actually GP119 increases. Now, from the behavioral point of view, if you take aged mice, in this case 16, these are young, in the Morris water maze, I think you know what it is, there is a hidden platform that is cannot be seen by the rodent, and then there are spatial targets that can be identified and learned by the animal, and you see within a training of 4 days, the animal goes very quickly to the platform, even if he doesn't see it, but he knows where it is. Now, in old mice, it takes a long time. You see they turn around. And here is when, during training, if the molecule is given during training, not during test, you see that the distance is much shorter, and you can see here the difference. Essentially normalizes in old mice the cognitive performance. Okay, sorry. In the ApoE4 model, which is available, same profile essentially. In ApoE4 vehicle, long time to find and distance that the animal has to take. But in the presence of GP119, it is decreased quite significantly. And in another model of Alzheimer's, the streptozotocin, intracerebral streptozotocin injection, which creates hypometabolism in the brain only, there is the same, a strong effect in decreasing, in improving cognition. And interestingly enough, very recently, actually now it's 3-4 years, that GLP analog, you know, everybody knows Ozempic and the others. It turns out that they have shown, it's been shown that they actually also stimulate glucose uptake into the brain by this more systemic effect. And in fact, a clinical study is undergoing phase 3 in Alzheimer's patients. And what was interesting to observe is that there is a synergistic interaction between GLP-1 receptor agonist and GP119. So mainly possibly allowing to decrease the amount of GLP-1 agonist to achieve this effect. Also in another model of neurodegeneration, ALS, you see here the progression, very rapid progression in muscle weakness in the SOD1 G93A model, and this is very much slowed, not stopped, but slowed by several weeks by administering daily GP119. So very simple take-home messages. Aging is characterized by hypometabolism. If one can target hypometabolism as a therapeutic approach, astrocytes are the main providers of energy to neurons, and so the targeting of astrocyte metabolism as a therapeutic approach should be considered to maintain cognitive function. And in fact, there is now molecules and some follow-ons that are clinical candidates that are well advanced in close to the IND, finishing the IND enabling and going to phase 1, hopefully by the end of the year. Thank you. Yes?
Okay, so you showed that it improves brain cognitive functions, right? GP19 were peptide molecule. What is the effect on? Does it affect obesity as well? The same molecule?
Let's say in the these were acute effects over and measuring glucose levels and lactate levels in the brain after acute administration of both GP119 and GLP-1. I think it was semaglutide. And in fact, very recently, new experiments show that you can give doses of the two molecules very low that have barely any effect on glucose utilization, and they synergistically act to increase.
Thank you. I would like to add this a movie scene. There was a movie. They do this spies. They catch the spies. They make them do sports very hard, produce large amount of lactic acid. When they are fast enough, then through those lactic acid memory consolidation, they try to catch their forgotten memories. Am I explaining myself? So it was a movie, but then later I heard that American Navy can do this. And right now it's so popular doing exercise in the morning, high intensity, and producing some amount of lactic acid to recover your brain. And the question part was, what about metformin?
Metformin, yes. Okay, first part, yes, indeed. I mean, exercise by increasing lactate, circulating lactate, but not only, because whenever you exercise, you also mobilize your motor and coordination neurons. So the astrocyte neuron lactate shuttle will be activated, and you produce also lactate in the brain. But lactate can enter the brain through transporters. And indeed, yes, there are several tests, actually, examples of improving cognitive function. Also mood, by the way, and I didn't show data, but actually we showed in a couple of papers in molecular psychiatry that lactate has antidepressant effects in animal models of depression. And so, yes, indeed. Now about metformin. So metformin, yes, it increases in vitro lactate production. We also showed it, and other colleagues of us in Germany showed it. In vivo, it's there, and also in vitro, it's much less effective than GP119 because the target is different. It's IMP kinase, as you know. Metformin mainly activates IMP kinase, and GP119 acts on another enzyme. Yes?
Thanks. Yeah, just my question, taking lactate as a supplement, does it restore the brain function and reverse the aging?
Well, that's a very good question. For the moment, we have worked on aged mice and to see whether cognition can be improved if they receive this extra glucose and lactate during learning, and it does, indeed. Now, whether we could, and that's the idea, to possibly start much earlier and prevent cognitive decline by taking these molecules much earlier. And by the way, I think the colleague here mentioned metformin. I mean, metformin is also taken even in non-diabetic people to improve also cognitive performance over time, or at least as a preventive. And not so much increasing, but preventive. And so one could imagine that this type of molecule that acts on the astrocyte neuron lactate shuttle could become the statins of the brain. In other words, take them preventively.
Thank you. I'm Dr. François Briand from Physiogenetics in France. Thank you very much for this very interesting talk. I have a question about the astrocytes, because you talked about the decline in glucose metabolism over time with aging. I'm just curious about what happens to fatty acid oxidation within the astrocytes and how it impacts the neurons. And also, do you have any information about the potential effects of ketone bodies?
Yes.
Thank you.
So definitely, astrocyte can produce ketone bodies, and they can deliver them to neurons. The efficacy, however, in terms if you measure oxidative phosphorylation in neurons, is lower than lactate. But they can be definitely an astrocyte-derived alternative substrate for neurons. In fact, the ketogenic diet is used for epilepsy, pharmacoresistant epilepsy. And most likely, and these are called metabolic epilepsies because they don't respond to GABA receptor agonists, for example, or other antiepileptics. And so the idea is the ketogenic diet can provide this extra substrate for neurons to maintain their excitability or control their excitability. Now, there is actually a disease called glucose transporter 1 deficiency syndrome, or de vivo disease. In fact, these are kids who have mutations in the glucose transporter 1, and GLUT1 is present in endothelial cells and in astrocytes, not in neurons. Neurons express GLUT3. And the ketogenic diet somehow works in these kids. Well, the problem is that they don't get enough glucose because they're deficient GLUT1. They don't produce enough lactate, but you can bypass this with a ketogenic diet, which, however, has the big problem of not being so easy to follow because you have to eat a lot of fats every day, and after a while it becomes really difficult for the kids. So I think, again, this approach of boosting the astrocyte neuron lactates, in fact, I didn't show this because we are in an aging meeting and GLUT1 deficiency syndrome is neonatal, so it was not exactly the same time in the life. But we have very positive results in animal models of GLUT1 deficiency syndrome. Even in organoids prepared from a patient that has GLUT1 deficiency syndrome.
Hi, Pierre.
Hi, Mo.
Thank you for this fascinating talk. So I have a question about the hypometabolism. And can you shed some light on the molecular underpinning of the hypometabolism during aging? I'm particularly interested in mitochondrial mutation.
Yes.
Is that playing any role in this process?
Yes. So, of course, you're right. I mean, this would be the ultimate target to understand, what the molecular mechanism. One, we know, and it's been shown in many papers, I would say pre-beta amyloid hypothesis, I would say overemphasis, but let's keep it for another conversation, that there is a decrease, as I mentioned, for the ApoE4 in glucose utilization well before any symptom, and a decrease in glucose transporter 1 expression in astrocyte and endothelial cells. So there is the reason why, I don't think anybody knows, but it's a fact, there is less GLUT1, and that probably explains why there is less glucose entering the brain. The other aspect, the end of the chain, of the metabolic chain, is the mitochondrion. And the mitochondrion, in fact, there is vast evidence of dysfunction of mitophagy and other quality control of mitochondrial function with aging. And so somehow the mitochondria become like cars that use oil in addition to fuel. I mean, they become less efficient. And so you have to provide more substrates to produce the same amount of ATP in neurons, independent of the fact that eventually it's lactate. But if you form less lactate because there is less glucose, the result is the same. And so the question is that it's actually a relative decrease of capacity. I mean, it cannot adapt, and there is hypometabolism. Now, how this reflects to glucose uptake, I think it's a very good question. I mean, this is, for the moment, I think there is no real explanation, but it's a fact at the molecular level. Yes?
Thank you so much for this talk. I have a question regarding neurodegenerative diseases. So have they found any link between any neurodegenerative diseases and hypometabolism, glucose hypometabolism in the brain? Have they found about a link?
Maybe I go close to, there is so much background noise. Can you repeat your question?
Yeah, my question is there any link between neurodegenerative diseases and hypometabolism, glucose hypometabolism in brain? Thank you.
Well, the idea, I briefly passed on this, if there is a hypometabolism, cells will be less, in general, they will be less resistant to different insults like oxidative stress, inflammation, inflammatory cytokines, and others. So the idea is that these cells, because they have less energy availability, they become, the processes that are energy dependent become less efficient, and so cells become more vulnerable. So the idea is that, so answering your question, yes, there is a correlation between neurodegeneration and glucose utilization. At least there are many studies on this.
Thank you. Thank you very much for a fascinating presentation. I really enjoyed it very much. I was wondering, dietary restriction delays aging.
Why?
In dietary restriction.
Yes.
Lower caloric intake. And that also protects against neurodegeneration and actually can revert to some extent neurodegenerative phenotypes. So how does dietary restriction link with hypometabolism in your model?
Yes. I don't think I have the answer, but what I know from the literature that one of the consequences, positive consequences of dietary restriction is a decrease in oxidative stress and inflammation. And so we are here in this balance that if you have maybe a deficient system in the brain to protect itself against these two factors, if you decrease the cause, in other less inflammation and less oxidative stress, maybe the available energy
net result will be positive.
Can match, yeah.
Yeah, okay, thank
No,
you.
I think it's positive, of course, yes. Yes. Yeah.
Just within the same lines of dietary restrictions, so fasting then, is there evidence that it promotes production of lactic acid?
What?
Is there evidence that fasting, for example, within the same lines?
No. What fasting produces, actually this is also interesting. Fasting increases the expression of monocarboxylate transporters in the blood-brain barrier. So you're more likely to use fatty acids and also, so you can use alternative substrates that use fatty acids. In fact, there have been old studies where they suggested, and I think even maybe some recent one, a ketogenic diet in brains that have hypometabolism due to insufficient glucose uptake. So you can provide alternative fuels like this. And so fasting increases the expression of MCTs in the blood-brain barrier, meaning that more fatty acid can enter the brain. Thank you very much.
That's all right. While she's getting the clicker, first, I'll tell you. I think this late in the afternoon, it's good to sort of think about the so what and why are we all here. And I realize that a lot of people who come to this conference are sort of just checking out the field of aging, but I fell in love with aging health in the 90s. And the reason that I got interested in aging health, oh, thank you. That was my fault. I should have asked. All right. There we go. So that's where I fell in love with aging health. So that is the residential memory unit of the Palo Alto Veterans Hospital. That's where veterans near Stanford, which is where I went to college, go with late-stage dementia to live and be cared for. So I volunteered there because I wanted to go to medical school and was looking for volunteer opportunities to be around patients. And the very first night I was there, thoroughly enjoyed my evening. So I have to say it was interacting with and being around older men with dementia. And on the way home in the bus, I was astounded to hear the other college students saying, I'm never going back there again. I'm getting a different volunteer job. I did not like that. I did like that. And it occurred to me that I had heard this quote in the past that your calling is where your great gladness meets the world's great hunger. So that night what I realized was that this was a great gladness for me, and it was really twofold. It was one, that I was a biology major, very interested already in the biology of aging. I was in a molecular biology lab. I was fascinated by what was going on in the brains of these individuals and what was causing what I was seeing clinically. Also, I really liked the population. I enjoyed being around them. And then later, in a class that was not a biology class, a sociology class, I began to hear about the aging population and the idea that over the next decades, this issue of not having enough people interested in the problems of aging, interested in the biology of aging, or wanting to take care of the population was going to be a major challenge facing the world. So I felt that I had sort of found this place where my great gladness met one of the world's great hungers, and I've been on this path ever since. My next lesson in dementia, and the thing that really got me interested in this too, is that this is me graduating, so that's a fresh-faced 22-year-old me graduating. My family had come from a small town where we lived in Arkansas to see me graduate. That's my grandmother. This was the trip that on this trip, we began to realize that something was wrong. Something was not right. We had thought before that sort of her changing cognition had something to do with she had lost my grandfather and maybe it was grief. On this trip, it was very clear that it was something more than that. We went home, she got the diagnosis, was diagnosed with Alzheimer's disease, and she died 9 years later. Very typical course of the disease. By the time she died, she didn't recognize any of us. So, what I know now that I didn't know then is that the changes that had been happening in her brain on that trip in California, those changes had been happening for decades prior to that trip. So this is an artist's rendition of the aging brain with Alzheimer's disease. What you see is the big balls that are amyloid. The blue represents the tau. Tau tangles inside the neurons. And that's probably what her brain looked like at the trip. We probably would have already been able to see amyloid and tau had we had the ability to look for it then. The other things that we know happen are loss of neurons, and that the microglia, as we heard in a talk earlier, become very activated. But what you're seeing over here is the time scale of these things. So again, the green lines, far out in time, are when we're seeing the cognitive changes. And what we see before that is the accumulation of amyloid beta that we can see first in the cerebral spinal fluid when we do a lumbar puncture. Then we begin to see the tau appear in the cerebral spinal fluid. PET scans become positive. The FDG PET, that shows the hypometabolism that we heard about in an earlier talk. And again, the detection of all these things is now years to decades before we see those cognitive changes. So, flash forward to now when I direct an Alzheimer's disease research center. And one of the things about Alzheimer's disease that has always fascinated me is that we know it's a highly genetic disease. People are born with those genes, and yet most of them live 5, 6, 7, 8 decades before even those changes that I was talking about before. So those changes are starting in people in their 40s, 50s, they're later going to manifest the symptoms in their 70s, 80s. But why are they born with these genes and they get 40 years without even manifesting the changes? So what I really think of Alzheimer's as is a disease of aging. There is something about having a genetic propensity, having the right sort of environmental factors as well. But what I'm very interested in is what are the factors that evolve with age across the lifespan that flip, that make the person with the right genetic propensity begin to manifest those changes in their brain that will ultimately lead to cognitive problems. That's what my center focuses on. So we focus on these factors in midlife, early, mid, and early late life that drive the change. So really what we're focused on is the phase of the disease even before amyloid is in the brain. And this is really different because most other Alzheimer's disease studies out there do what you think you'd do if you wanted to study Alzheimer's disease, which is enroll a bunch of people with apparent Alzheimer's disease. So they tend to enroll older adults that have symptoms or high-risk, rare variant families, that family AD where it manifests earlier than the typical garden variety Alzheimer's disease. And most of the people that have enrolled in those studies have been highly educated, high socioeconomic status, advantaged, mostly of European ancestry. So you'll hear in a little bit about how we're sort of trying to flip all of this and be the first Alzheimer's disease center that is enrolling people who don't have Alzheimer's disease and look a little bit different than some of the other populations. All right, but I want to tell you too that, remember, I am a geriatrician, and a lot of what I worked on before even running an Alzheimer's disease center has to do with resilience. So somebody asked earlier today, how do you define aging? What is aging? And I thought that Alan Cohen gave a good example, a good definition, which is it's the loss of information. I sort of think that you can sum up a lot of aging as just a changing capacity, a declining capacity to respond to health stressors. So loss of resilience due to the loss of information or the loss of information within a complex system. This is the framework at Duke that we use to study resilience. One of our centers at Duke, the Pepper Center, focuses on physical resilience or health resilience to many different kinds of stressors. This is intentionally a very simple model. It's because we apply it to a lot of different stressors and a lot of different populations. But we try to measure first what is a person's pre-stress reserve. You could also call that intrinsic health or intrinsic capacity. How can we characterize their health before they get stressed, characterize something about the nature of the stressor, the magnitude of the stressor. Then what we focus a lot on is what I see as the dynamic process of resilience. So how the system responds at that moment of stress. Usually that is an adaptive, plastic, helpful response, and very often what we see in aging is it starts out that way, but then it's not regulated in the way that we would want it to be to continue to mete out the good outcome. Much of successful aging, I can tell you as both a clinician and a researcher, much of successful aging has to do with how many stressors and what stressors a person encounters and how they respond to them, how resilient they can be to them. So I really see those as major opportunities to intervene at a population level. And I'm not going to belabor this because we've been hearing about it all day long, but we certainly ascribe to the science of dynamic resilience. And I am applying that to how I think of the aging brain. So I think of the aging person as a complex dynamic system that you could break down into multiple subsystems at multiple different levels. But importantly, all of those subsystems are cross-talking to each other, feed forward, feed backwards. And then, of course, an individual also lives in another complex dynamic system, which is society. And in that, we have the brain that I focus on a bit, but also I think of the brain as part of this complex dynamic system, affected by and affecting the whole rest of the system. And what I'm very fascinated is what is it, again, in the person with the right genetic makeup, that when this system gets off kilter in just the right way, we begin to see those stereotypical changes associated with Alzheimer's disease. And, I think of all of this as sort of a vicious cycle. So diseases and injury, they diminish our biological resilience at that molecular cellular level. So when we get injured in life or when we acquire a chronic disease, it chips away at our biological resilience, and that lowers our ability to stave off the next chronic disease. So this is why this is a red vicious cycle. The good news is geroscience, the rainbow in all this, is that molecular nature of resilience has a biological underpinning. And much of what we've been talking about today is the opportunity to intervene in these sort of hallmarks of aging, pillars of aging pathways that should be able to restore a more youthful resilience to the whole organism. So that's a lot of what sort of drives our approaches. So what does all this have to do with the aging brain? So again, an artist's rendition of a healthy brain now. And what you can see is that the neurons, constant dynamics, we've talked lots about that today, talking to each other, but importantly, talking to other cells too, as we just heard, astrocytes, microglia, and then the blood vessels. So all of this, the blood delivering good, healthy things, oxygen, also delivering potentially toxic things, bad things. And so a lot of my work is to try to understand how this system, which is in a constant state of dynamics and seeking a homeostatic equilibrium, can be perturbed in very specific ways that can lead to Alzheimer's disease or can lead it to not respond well to the changes of Alzheimer's disease. So first, I think probably many people are aware of this, but dementia represents this very heterogeneous mix of pathogens. So because I'm focused mostly on Alzheimer's disease, I think about amyloid and tau, which are the two misfolded proteins that are characterizing Alzheimer's disease. But of course, amyloid and tau exist with other proteinopathies that we now know about and can characterize: alpha-synuclein, associated with Lewy body dementia, Parkinson's disease, and TDP-43, which is associated with late, or one of the newer classified common causes of dementia. And interestingly, all these things often co-occur. So if I follow a person who has Alzheimer's disease, I take their brain at death, look at it, what I'll often find is the amyloid and tau is there, sure, but it also has some of these other misfolded proteins as well. These things very, very infrequently are pure, even though we classify the diseases according to which proteins are there. Now, this is the stress and resilience part. What I also sort of think about with the accumulation of these proteins and the proteinopathies is that the brain is constantly exposed to other stressors too, of a variety of different kinds. So it has to deal with changes in blood flow, changes in its oxygen delivery and the other things that blood should be bringing it at different times. It has to deal with inflammation and immune responses to pathogens, certainly, but to other things as well. It has to deal with its altered cellular metabolism. We know some of that is age-related, but it may be that some of that is disease-related too. Increasingly, what we're seeing is that some of the genes that are associated with Alzheimer's disease also affect cellular metabolism. And so it may be that there's something specifically about altered cellular metabolism that causes those proteins to begin to become misfolded, extruded, and accumulate. And then, of course, brain injury, so traumatic brain injury or ischemic brain injury. And all of these things, so we have a system that's constantly trying to keep itself healthy. It's experiencing the normal stressors of anyone's life, but as it ages, its ability to sort of fend off these stressors is declining. All right, so I'm going to just give you an example of two of the kinds of studies that we do in our center. I could have chosen from lots. It's a big center. But I wanted to give you two examples because they sort of tap into different kinds of resilience. One is this idea of cognitive resilience, and how I'm sort of defining that here is the mind's ability to adapt and perform cognitively, to continue to remember things, to have language. The mind's ability to adapt and perform despite the presence of some kind of a structural change in the brain. Then biological resilience, which is really like the brain cells' ability to respond to stressors at the cellular or molecular level, and thereby resist tripping into this neurodegenerative process and ultimately cell death. So I'm going to give you just an example of each. We'll start with cognitive resilience. So, what I'm showing you here is the Lancet Commission recently, or this is now the second time they've done it. They did it first in 2018 and recently updated. These are sort of factors across the lifespan that have each been linked to the risk of dementia in epidemiological data. So associatively speaking, these are things that are all strongly linked across multiple populations and in meta-analytic studies to the risk of dementia. And what's interesting is if we look at the population attributable factor of dementia in these studies, if we modified these 14 modifiable risk factors, we'd be preventing or delaying up to 45 cases of dementia. So this in some ways is a pretty good template to look at if you want to start trying to prevent or stave off this disease. So the first one I'm going to talk to you about, again, this comes from my earlier work. I'm a geriatrician. At that time, most of my clinical studies were in older people. And so I was looking in the later life part of this path and very interested in this relationship between vision and dementia. So we know that sensory impairments, both hearing impairment and vision impairment, are associated with reduced cognitive function and dementia later in life. And there's lots of potential things that could account for this association. We don't know exactly what causes it, but all of these are possibilities. And one possibility that I was really interested in was this idea of brain structure and function, that something about losing your vision in late life could actually change your brain structure or white matter in a way that was a challenge to this aging brain, a stressor to the aging brain. Several studies also had shown that particularly people who develop age-related macular degeneration are at increased risk of dementia, and even before they get dementia, they underperform on some very specific tests of verbal fluency. So verbal fluency are tests where I might tell you, in 1 minute, I want you to name all the animals that you can think of. And that's not the task that should cause you to rely on your vision, so it shouldn't matter that you're legally blind, that you underperform. But people with age-related macular degeneration begin to underperform on that task. So I was very interested in understanding sort of why that would happen. I had a little bit of a hypothesis about why it was, and I'm going to demonstrate it to you here. So we need some audience participation. So we're going to play a game where I'm going to show you some photos, and each time you see a photo, I want you, if you recognize the person, to say the person's name. I'm from the United States. They are all Americans, but I think you might recognize some of them. Bill Gates. Taylor Swift, that's right. All right, so congratulate yourselves, those of you got those, and just take a minute to marvel at what your brains just did to make that happen. So you see the picture, and that information goes from your eyes, through your optic nerves, to your laterate geniculate nucleus. It goes through your splenium, radiates out, goes to your occipital cortex. And once it hits the back of your brain, the occipital cortex, it begins to understand the dimensions, the color, the contours. All of that happens in less than 200 milliseconds. And then over the next 200 milliseconds, that information fires back up to the front of your brain where all the other lobes light up like a Christmas tree, and you start to make sense of this. What does it mean? Do I know the name of this person? Am I afraid of the person? Do I like the person? Am I attracted to the person? All sorts of split-second decisions are made, and then you have to involve language because you have to speak the name. So to do that, your language, which is a bilateral, you can see the language network lit up down there, especially on the left side, those two come into activation when you speak the name, and in 400 milliseconds, you've said the name. So all of this becomes a little slower with aging, and people get a sensation sometimes of tip of the tongue. That's normal aging. But interestingly, this process goes on your whole life, not just when I do it arbitrarily here, but when you walk around here and you might see someone you know and you immediately say their name. So this is a pathway that gets triggered over and over and over and over. And now imagine that we take away your vision late in life. And so you can imagine that the lack of that sensory input is in fact a major stressor to the brain, decreased activation, decreased stimulation. And what I wanted to know was what exactly is changing in the brain when this happens? Does something structural change that I can measure? And are there some people who seem to resist it, who seem to be resilient to it? So we enrolled 81 people with age-related macular degeneration and then 85 age-matched controls. We did a lot of neurocognitive testing. We did a lot of ophthalmologic testing and characterizing their condition. And then we did brain MRIs in people who were eligible. So we had, many of these people have something like a defibrillator or a pacemaker because the average age is 75. So many of them can't have an MRI, but those who could, we did an MRI. And I'm just going to show you some of the things that we found. Since I know a lot of you may not be used to looking at MRIs, I'm going to tell you what we found first, and then I'll show you the picture. So first of all, what we found was that the AMD group always had worse white matter integrity in certain tracks, and those were tracks that are associated with language. I don't know if, so had that image played, it would spin the brain around, and it looks really cool. But what you can see is that there's a little horseshoe track there that is the splenium, which is kind of a superhighway of information going from that occipital cortex to all those other lobes. And what we see is in the people with age-related macular degeneration, their white matter integrity was worse at baseline than the other folks. Now that could, we thought, be maybe it just means that they're people that have worse white matter integrity, and something about that made them be at risk for getting macular degeneration. The other thing that we found, though, was that not only did they start out with worse white matter integrity, they declined faster. So they had more deterioration in those white matter tracks, and it was especially on the left side, which is the main language sort of prominent side. So what we found was that people, again, that lose their vision late in life, and these were all people that were legally blind from age-related macular degeneration, their brains seemed to be sort of aging faster, and particularly on the side where language is involved. And then what we found that was really interesting, though, is that we did see that there was this sort of connectivity pattern. So now we're not looking at the structure of their white matter tracks, but we're looking at how different brain regions that are separated from each other, how they connect and talk to each other. And what we found is that some people that had a particular connectivity pattern were more cognitively resilient if they had AMD. So people who had AMD that had a pattern that looked like this had more activity on the right side of their brain, talking to the other parts of that language network. Those people who had AMD, despite having AMD, despite having worse white matter tracks, were still able to do well on the cognitive tests. So what that tells us is that that is sort of a resilience mechanism. So the question is that something that we could create for people? And that's a question that is yet to be answered. But finding this idea that, yes, the mind has abilities to be resilient, to perform despite structural damage that we can see from age-related stressors. Now I'm going to give you one more example. This one has to do with hypertension, and we think a lot about the vascular factors in the brain and how vascular risks across the lifespan are impacting our aging brain. All right, so first, this is a patient, retired statistics professor, comes in with 2 to 3 years of subjective executive dysfunction, memory loss, classic dementia presentation. Again, this man is 79 years old, and these, you can see his MRIs, and what he has is significant vascular burden. So he didn't know that he had had strokes. These weren't strokes that cause obvious neurological symptoms, but when we look at his brain, he's had several strokes and quite a bit of random stochastic vascular insults. So this is a case of vascular dementia. What I'll tell you is this is my father-in-law. This is why I can show you the picture. He's given permission. And this is my husband. So knowing his dad has vascular dementia, I'll tell you, when he was 43 years old, there was one day that I was driving home from work and we were talking at the end of the day, as we often do. And he calls at the end of the day and says, can I just ask you something? He's not a doctor. He said, one side of my mouth has been funny ever since I went to the gym this morning. I said, what do you mean? And he said, it's drooping, and I'm kind of drooling on that side, and it's been doing it all day long. So I said, okay, stop. We're not taking the kids to karate lessons. We're going home. I'm going to take a look at you. We'll see what's going on. And to me, and again, I don't know if this will play. Oh, yeah.
My name is Ben.
Now, it's very subtle, and you might not see it, but on the, let's see, left side of his face, yes, The nasolabial fold, this thing right here, is just a little depressed compared to the other one. Many people didn't notice it during his day at work, although a few did and commented on it, but I saw it immediately. And so we went straight to the hospital, and he had had a stroke. So he had a very small punctate infarct. This is a subcortical ischemic stroke. Now, the nice thing is he's 43 years old, and I'm absolutely positive that if he weren't married to a brain doctor, he would have gone to bed and woken up the next morning. And he's almost fully recovered, to be honest. By 2 days later, he still has numbness that persists to this day, but the rest of it was gone. I was able to see his brain. It's a pristine brain, except for that one little punctate area. But what this tells me is he's on the path of his dad if we don't do something about it now. So at this time, he was 43. He's now 50. He's a fitness guy. He does a lot of CrossFit and things like that. This is him doing upside-down push-ups for the entertainment of our children. So some of the things that he started doing, he had always been sort of attentive to diet and exercise. He really got more attentive. He gave up cigars, and a lot of medications got added. So is that ideal for a guy in his 40s to have to take a lot of medicines? No, but is it better than ending up with dementia by age 79? Sure. So this is, this motivates a lot of what we do in the center, which is really trying to understand what are the other options, what are the things that we can do for people in their 40s who are starting to experience brain challenges and stressors, some that may be asymptomatic and they don't even know yet. At Duke, we're the coordinating center for the preventable trial, which is looking at statins as a potential preventive indication for dementia and cognitive impairment. They're obviously widely used for hypercholesterol management and to prevent other diseases. But we have lots of other studies going on that target sort of these other factors. And a lot of my questions, because again, I'm not really an interventionalist. I study mechanisms and biomarkers. And what I'm concerned about is that I don't think we have the data in the right population yet to answer the questions that we really want to know about prevention. So this is what we're doing. We have 2 cohorts going on of these deeply phenotyped humans with a family history of Alzheimer's disease. So these are people who do not have cognitive changes, but they have a family history or a known ApoE4 gene. They're representative of the population of Eastern North Carolina, so about 20% of our cohort is of African American descent. Many of them come from rural areas in North Carolina because we know that the exposome is really important. The memory and aging study is people ages 45 to 80, and we follow them longitudinally every year until they die, and we will collect their brains. And then the young cohort is people ages 25 to 44 who come in one time until they age in. And the reason that we don't see them every year is it's just not economical when we don't expect a whole lot to be changing between age 25 to 44, but we want a baseline. We collect a lot of data, so we get detailed cognitive data, retinal imaging, high-resolution brain imaging, sensory and mobility measures. We store their plasma, buffy coat, DNA, and all of them have either consented to a cerebrospinal fluid test, so a lumbar puncture, and we bank that, or PET images. This, again, as I said, this is the main cohort that we follow. Up to 100 of them can be found when they come in to have mild cognitive impairment or even early dementia. They don't come to us saying that they have that, but sometimes we find out that they do. But we're not allowing any more than 100 people at any given time in the study that have symptoms. The rest cognitively normal. And then we have this one if they're a woman between the ages of 45 to 60, we collect additional information about them to characterize where they are in menopause. All right. And as I said, if you go to that QR code, you can see online. We put all this information online, very into open science. We collect this to share it. I think that the best biorepository is an empty biorepository. So we really want this data to be put to use because our participants want it to be put to use. These are an incredible group of people who are cognitively normal and yet agree to all this invasive testing and time spent with us because they have a family member that had this disease and they know how bad it is. That's the motivation. All right, so I think that, as I said, I've been in love with aging health and aging brains for 20, 30 years now. But I think now is the time, partly because the population is aging. We're going to see more and more and more of this, but also because in those 30 years, we've really accelerated our ways to stress the brain. So although brains have been aging for a really long time, our modern life has created all kinds of new stressors. So all the screen time that we use, unprocessed foods, climate change, all these things as yet to be determined what this means for those factors across the lifespan. I'm standing here before you as one person, but I obviously represent many, many people, even more than are pictured here on the slide. It's kind of too many to picture. We have 2 schools, Duke and UNC, 11 departments, one common goal. I will end there and be happy to take questions. Sir.
Can you pass this?
Hi, that was great. I'm just interested to know, I saw on your list of the determinants of Alzheimer's, it's a long list, and many of the factors. And when people say 45% reduction, it's almost as if many of them are very easy to reduce, but they're actually pretty tough.
Yeah.
But I just wondered how that list compares to whether you've looked at the Lothian birth cohort from Scotland, where they followed them, for 60, 70 years. And there they were looking at determinants of cognitive decline. If I'm right, they were a very similar list. And now they've got all these that, as you know, maybe good just to hear you speak about that cohort, because that also seems an extraordinary resource for addressing many of these issues.
Yes, I completely agree with you that the birth cohorts. So first of all, I suspect that data probably informs that list that I showed. The list that I showed is not my list. It's the Lancet Commission's list, and they certainly incorporated epidemiological data from lots of longitudinal studies. So I would be surprised if the Scotland study is not in there. Other birth cohorts from England, New Zealand, other places. And then I forgot the second part of your question. Yeah, so the answer is yes, that they're certainly related, and because Alzheimer's disease is the most common cause of age-related cognitive decline, it's sort of like the most common cause of the cognitive decline. There are others. So it may be that there are some things that cause cognitive decline but don't cause Alzheimer's disease. One example of that would be vision impairment. So I can't think of any particular way that losing your vision causes you to develop amyloid and tau. I could be proven wrong, but I don't think so. I think what it causes is a decrease in your ability to respond adaptively to having amyloid and tau in your brain, or other insults to your brain. I was going to say one more thing about the Scotland cohort. One of the problems with the existing data that's there, because those are such rich data, part of the problem is what they don't have often is what I need with regard to the brain. So many of those people didn't agree to lumbar punctures, so I don't have their CSF. They didn't have PET images. They didn't have retinal images. You know, there's new ways of sort of assaying or getting at what's going on in the brain. What they did have often was banked blood, and we're getting better and better and better at being able to detect blood changes that correlate really well to PET imaging changes. So I think the use of those birth cohorts is about to get way more useful for studying Alzheimer's disease.
Hi.
Yes.
Could you comment on the role of brain atrophy in cognitive decline, and do you think that interventions to prevent or preserve brain architecture would be effective in maintaining cognitive function?
Yeah, so I think, atrophy really just means shrinkage of the tissue, right? And lots of different things could be causing it. Certainly, some of it is just age-related. We know that there's age-related loss of the tissue. But we also know that these neurodegenerative conditions accelerate age-related loss of the tissue, and we see worse atrophy. There's also the question of, is it global atrophy? All the lobes are kind of shrinking at the same rate, or do we have certain lobes that are shrinking faster than others, which usually suggests that it's one of these neurodegenerative conditions and not just age-related stuff. But regardless, things that protect the nerve cells themselves or their myelin sheaths, those should help preserve cognition. So yes.
Hi, great presentation. Thank you so much. So obviously, besides all those risk factors, we know the common ones. Just a couple of days ago, microplastics got highlighted, deposited in brain. Amazing study.
Yeah.
Are you collecting microplastics? Which level are you collecting in these patients? Can you shed some light on that?
Yeah, so speaking of one of those modern life creating new stressors for our brains. So yes, that we think about a plastic spoon's worth of plastic can be extracted from most brains around the world is terrifying. We actually don't know how terrifying it is because we don't know what damage that's causing, but it doesn't sound good, right? To think that there's these microplastics in our brains. And that's certainly something that like my grandmother wouldn't have had in her brain. So, it's worrisome. We are not specifically right now funded to assay or look at microplastics. But the nice thing about this cohort is that we easily could be. It's an incredibly easy thing to add on to these people that are coming in every year and seeing us anyway. And they expect that. These participants are asking for that. When they come back in, they always say, what's new out there? What new test are we doing today? What is the research pointing at that we should be collecting about me now? Because I should say, too, we share all of our information back with these participants. That's another big motivation for them.
Yeah, sorry.
Yeah.
Very interesting observation that you mentioned was that age-related macular degeneration is actually a predictor, at least can be a predictor for Alzheimer later on.
Yes.
So macular degeneration occurs in the retina.
Yes.
So there are different neurons involved.
Yes.
So how do you explain, this very different location
Yes.
and a very different functional state of neurons
Yes.
be connected with Alzheimer, which is more in hippocampal?
That's
driven?
right. That's right. No, great question. So I've looked a lot at the available data on this topic because I think it's fascinating. So I think one possibility is that some age-related, or let me say it this way, some aberration or genetic risk in some age-related pathway puts you at risk of both. So you're right. Although it's different neurological tissues, it's two neurological tissues, and in both cases, part of the problem is accumulation of sticky proteins outside the neurons. In macular degeneration, we call it drusen, and they're amyloid-rich, but not all amyloid. In the brains, it's these amyloid plaques. So there's similarities between the two, and it could be that some fundamental mechanism links both of them. If you have a genetic propensity to be someone who, when your nerves are stressed, they extrude sticky proteins, it may put you at risk for both. The other possibility, I think, is that some of the studies, when they were diagnosing the people with Alzheimer's disease, it was back in the days when it was really hard to detect amyloid and tau in a person who was alive. So it was a clinical diagnosis of Alzheimer's disease. And what they may have actually had was any form of brain problem, and the fact that they couldn't see was stressing their brain in another way and making them look like they had dementia.
Uh-huh. But so do genetically predisposed Alzheimer patients have already...
Great, another question. Okay, so ApoE4, the gene that is most associated with Alzheimer's disease risk, if anything, there's one study in humans that looks like ApoE4 was protective for age-related macular degeneration.
Mm-hmm.
That is crazy, right? That blows it out of the water that that these two diseases are actually linked. Except it's possible that it was a selection bias because the people who were ApoE4 carriers already had Alzheimer's disease by the time they might have gotten age-related macular degeneration. So they were sort of not presenting for studies about age-related macular degeneration because they had such bad dementia. So it's possible that it's a selection bias that led to that observation. But if anything, the observation is that people with ApoE4 looked less likely to show up in AMD studies.
And finally then, the ones who have age-related macular degeneration before,
Yeah.
do they end up in a subset of Alzheimer patients or certain subcategories of Alzheimer patients, or are they overall more, so you show the heterogeneity and you show the overlap, so if you put those patients on that picture, are they a subset or are they general?
Yeah, it's a great question, and we don't know. Because for so many of them, part of it is, this is the problem with siloed medicine. The ophthalmologists were studying age-related macular degeneration, and the neurologists and geriatricians were studying Alzheimer's, and nobody was collecting brains and eyes at death and looking at both.
Thank you.
Terrific presentation. Do you and your the UNC Duke project have any collaboration with Dr. Mia Kivipelto in Sweden and her worldwide fingers study?
I don't think so. Say the name again.
Mia Kivipelto. It's the lifestyle study. It's
Okay.
actually a big part of the Lancet report.
Yes, so I'm aware of the lifestyle study, but I'm not familiar with those investigators. We work with a large group in Sweden on blood-based biomarkers of Alzheimer's disease, yeah.
Thank you. It was a wonderful talk. I do have a question. My question is that with all the studies that are happening now in the center, with studying people, healthy population without Alzheimer's disease, do you think in the future are we going to have like a screening program to prevent Alzheimer?
I hope so. I mean, I think that is really the hope of all of this. Now, right now, we're deeply phenotyping these people to the point that no one would submit to these kinds of tests just coming to their doctor. I mean, they truly come in 3 different days, and all told, it's about 7 hours. The baseline data collection takes about 7 hours. So what I really hope is that we'll get to a point where we will realize what we need to collect and what we don't. We're already realizing some of that. Some of this testing is redundant. So in our next iteration, we're cutting out some of the tests because they're so related, we don't need to collect both of them. That's the hope, is that we hone in on exactly what's giving us the most information that we need to test. And that's exactly what we hope. Now, again, the problem with enrolling people in their 20s and 30s and 40s and 50s is that it's going to take 30 years. I said I've been at this for 30 years. I think I have another 30 in me, but I hope so. It's possible that I'm sort of planting seeds under the trees. I will not sit under their shade, that it'll be someone else that needs to carry this on, because I think the real payload for a cohort like this one, like with all the birth cohorts, will be decades down the line, but that is exactly what I hope.
Thank you.
The flippant but serious comment for you to reflect on is I've always studied cohorts, and the longest cohort still going in the UK is the British Doctors study of the 1940s. The good news is that the principal investigators almost always outlived their cohorts, even they went into the 90s.
That's good. Thank you. Yeah, we always say that a great way to have a really long, happy life is to enter a brain donation program, because we're collecting brains from people in their 100s that, again, who were enrolled not by me, but by people before me.
Thank you, Doctor, for this wonderful presentation. I have a comment and a question. Regarding the comment, as you have rightly mentioned, whenever there are some stressants, they really affect how the brain works, and it tends to increase or speed up these things.
Yeah. Mm-hmm.
I'm from Sudan. I have witnessed, due to the unfortunate war happening, how a lot of elderly of our relatives and of our population have slipped into dementia and into depression and into other things. Mm-hmm. And especially in one relative, he had a certain type of routine, daily routine every day, how he does everything, and he was doing fine. Mm-hmm. Due to the war, he had to relocate. His routine was disrupted. He immediately slipped into this situation. The question is, and here I'm talking as a caregiver, care provider, yes. And I mean for a patient rather than a physician, in layman terms. My relative has been taking statins for 30 years or 40 years, mm-hmm. Okay? And in the dementia, in the investigations, we have seen brain atrophy, etc. And there were talks that these statins worsen the situation. Now, what we did, we went to a cardiologist and we took permission, especially that my relative is around 83 years old. We took permission to take him off these statins and focus on giving some omega, supplements and dietary on fish, etc. Honestly, we have seen a very improvement, surprising improvement in his condition. He is an author. He went to a level where he would not be able to write, and now he's regaining his writing, his memory, and talks with the family. So could it be that some medications, especially statin groups, can impact negatively such?
So certainly medications are often not good for aging brain function. As a geriatrician, I'm usually not the most popular person at industry meetings because we stop more medicines than we start in general. But statins, it's an open question because of exactly what you said. There are some thoughts that acutely they seem to interfere with some people's cognition. Not everyone, but for some people they seem to have this as a side effect. But there are lots of biological mechanistic reasons to think that they may be protecting the brain in the long term. So the preventable study is the first large randomized controlled trial powered to look at exactly this question, which is if you take older people on a statin, specifically how does it impact their cognitive outcomes in late life? That question is still an open question, and that study's ongoing. As I said, Duke's the coordinating center. I'm eager to see what they find.
Thank you very much. Thank you.
Thank you very much for the presentation. In the beginning of your presentation, you mentioned that you actually measure pre-stress reserves. Can you talk more about it? What you measure?
Yeah, sure. And I'm going to give you some answers both from the study that I showed you about the young cohorts that we're looking at Alzheimer's disease risk, and also some other studies that we've done where we take an elective surgery as the experimental stressor. So we have a person that we know is coming up for an elective knee replacement. We collect a lot of information about them before and then information after the surgery. And so I'm very interested, and this gets a lot to what Alan Cohen talked about earlier, sort of that capturing that intrinsic health before a person is stressed. So trying to get a snapshot of their relevant health before the stressor, right? So what I think is the best way to do that is by stressing them. So as was mentioned earlier, I think on the main stage this morning, maybe it was Felipe Sierra who said, it's unethical to stress an older person so badly that you risk making them decompensate. But we have to find ways to expose people to little stress tests and get a sense of how much capacity they have. So some of the things that we do are dual tasking, where you make them do two things at once. Usually when you make anybody multitask, they perform a little worse on both tasks, but if they really fall off on one or the other, that can be evidence that they don't have much reserve in that system. The other thing that we do is we draw their blood and we stress the white blood cells. So a PBMC stress test is a good way to sort of stress their immune system without stressing the patient. And then we do other things like I have one test that we do where it's kind of like a poor man's functional MRI, but we use electrodes, and what I'm trying to get is their cerebral vascular reactivity to stress. And so it's an FNIRS that just takes two little probes in a dark room, and what I do is I look and see how quickly their oxygenation responds when I make them do math problems or when I make them stand up and they have a blood pressure drop when they stand. How quickly does it return? So those are the kinds of gentle stressors that we try to do. But I think that's one of the hottest areas of study for this field, is figuring out what exactly are the right biomarkers that are reliable, consistent, can be feasibly and safely and inexpensively measured, and importantly, that change with intervention or with stressors.
Thank you, Heather. One question, I'm really interested by this idea that structural aspects of the brain could be buffering or maybe anti-buffering some of these effects. And the idea of education as cognitive reserve is well known. Have you been looking at kind of some of the specifics of the tasks these individuals do with their brains, they're good at, that they like, how they're engaging with the brains, and then trying to relate that to structural aspects and seeing whether you can find anything there?
That's a great, so getting at sort of those social determinants of health and what these people do. So we certainly collect their employment. I don't think that we're doing, and we collect some questions about their physical activity levels. I don't know that we're doing as good of a job as we could with all of their hobbies and different things. And but I will also say, you know, right now we're only funded to do 420 people, so I think that that would probably introduce a level of variance that we are not powered to examine.
Thank you.
Thank you for your presentation. What's the state of sex-specific management of Alzheimer? Because we know the hormonal imbalance, especially
Yes.
postmenopausal.
Yes.
So is there any management specific for each?
Yeah, so great question. So in our study, and this partly came because you guys may or may not know this, and it may not be the same everywhere, but in the United States, women are way more likely to volunteer to be in observational studies than men. If you know any men, encourage them to maybe be in a study if somebody friendly calls and asks them to be. So at first we were thinking, you know, our cohort is 2/3 women, like we've got to like get some more men. And then we realized, actually, you know, we need to know more about women and Alzheimer's disease and perimenopausal. So we've added a lot of information that we collect that is both biological information, blood draws that do hormone levels, as well as questionnaires that get at menopause symptoms, so that we're characterizing the women who are 45 to 60, where they are in the menopause process, and that can become one of the variables that we'll look at. You know, as to the, I'm probably not the best person here. Dina Dubal is here at this meeting, and she's one of the world leaders in sort of sex-specific causes and contributors to Alzheimer's. So I would say find her.
Hi, thank you for coming. I want to know about a lot of the supplements that are on the market right now that are promoting biohacking and like slower aging of the brain. We see a lot of NAD supplements on the market. I don't really know if it's marketing or is it really some testing that supports it being successful for this?
Yeah, thank you for that question. So that is one of the most common questions that I get in clinic is, you know, I saw on the internet this thing, or I've started taking this big bag full of stuff of curcumin, and, you know, and the answer for a lot of those things is that often what it is there might be some preclinical evidence that suggests it's a helpful pathway. There's not often powered, consistent, randomized controlled trials in humans that demonstrate that it's effective. But you're right, because again, I mean, there is such a market interest in giving people things that they think will help them live longer or help them live healthier, or not get Alzheimer's disease. There's a lot of misinformation out there, and I have one colleague that I practice with, a neurologist, who will tell her patients when they bring up that giant bag full of, you know, every micronutrient under the sun and NAD and all kinds of stuff, and she'll say, this is making your urine very expensive. I don't know if it's doing anything else. And so, you know, again, the day might come where we have that data, and a lot of them probably aren't hurting people, depending on what they are, but it worries me a little.
Not so much a question, but a response to that. There is a wonderful website called cognitivevitality.com. It's run by the organization, the Alzheimer's Drug Discovery Foundation, and it has PhDs that look at all of these supplements and can tell you, they rate them by 1, 2, or 3 brains. 1, is it safe? 2, are there any studies out there? And 3, there's a study out there that actually proves a real connection. And it's just a good clearinghouse for these very questions.
I'm going to get that site from you,
you
because
got it.
I'm just going to start sending it to people.
You got it.
Hello. So you talked about stress and its relationship to Alzheimer. So have you found that patients with dementia and psychological disorders like major depression and major or general anxiety disorder show an earlier onset of dementia? And have you found any improvement in cognitive function of patients that managed their psychological disorder, like with SRIs or SNRIs?
Yeah, that's a great question, and it's really a personalized medicine question because it can be hard to tease that out. Because there are neuropsychiatric symptoms of dementia that are not sort of the same as depression outside of dementia. And depression and dementia or Alzheimer's disease are both so common that a person is allowed to get both, can get both of them. And it's not that one caused the other, they just got both. So there's a lot of work now in this area of NPS, or neuropsychiatric symptoms of dementia, and often they will look at anxiety, depression, apathy, agitation. There's kind of a whole list of them. In the teaching is that when it is a symptom that is of the dementia, depressive symptomatology usually doesn't respond to the SSRIs, whereas when it's depression that's not a neuropsychiatric manifestation of the dementia, it does. And so what I usually do is do a trial and hope for the best, and hope it's a symptom that will respond to the SSRI. And if it doesn't, then sometimes we know it was probably just part of their dementia, even Alzheimer's. Even when it's very pure Alzheimer's, at death, the person really does seem to have just Alzheimer's-like pathology in their brain. Still can manifest really, really differently. It just speaks to the fact of that difference between sort of intrinsic health and realized health. Many other factors determine our sort of cognitive phenotype, if you will. All right, thank you very much. Thanks.